Good morning, everybody. This is Schmidt Ocean Institute, Research Vessel Falkor, and ROV SuBastian. We are just starting our live stream. We just got to the seafloor. We are at 2,417 meters. Doing a small setup, white balance. We thank you for joining us today. Our science team will join us shortly and introduce themselves. But we just want to let you know we are streaming on YouTube and Facebook. You can find out more about this expedition at schmidtocean.org website. Go to cruises. But we are currently still at Baker Island in the remote Pacific Remote Islands Marine National Park Monument. And we are just up on a ridge to the north side of the island. So we dove on this same feature yesterday. We're just at a slightly different part of the island. So thank you for joining us. Come follow along. Send your questions in YouTube and Facebook, and we'll try to answer them. Thank you very much.
Morning to everyone from, wow.
Is that big enough?
We want something more substantial, like 15, 20 centimeters. Yeah. We're looking for something that, has some weight to it a bit. It's not going to disintegrate.
Yeah, that thing kind of disintegrated when you touched it.
It doesn't have to be right here. It can be in the near future too.
See if you can pick that up. Yeah.
So as I was saying, good morning everyone. We're diving just shortly after our last dive on the southeast corner of Baker Island, but instead this time we're on the complete opposite end of the island on the northwest at a depth of about 2,400 meters. So while we were in the shallows this morning, we're now much deeper, trying to explore some of the substrate and fauna communities in this part of the seamount, or island rather. We're still at Baker Island. If any of any of this stuff is, if it's breakable, is also good, but like if it's loose, that's less work. We are on the search for a rock, kind of our first order of operations when we get down to the seafloor here. We've been collecting several rocks through the course of our dives over the past few days. These rocks serve a couple of purposes. Namely, we're trying to figure out a couple things. First being how old some of these underlying substrates are on seamounts and islands in this area. So we've been collecting some volcanic rocks. That might be too big for the quiver, or for the rock box, you think? Yeah, it's pretty. Any of this, like, we can even break stuff off. I don't know if any of this is. We're looking for basically a good sized chunk. I mean,
There you go, right there. Oh,
yeah. Is that yeah, completely? Can we poke that again? See if it totally falls apart.
The big one?
Yeah, the big one. I'm just curious. Yeah. Okay.
You want
That.
Can break it again?
No. Can you grab that piece right there? That. Yeah, you're pointing at right there.
Okay.
And
I'm gonna try and roll it my way a little bit. Okay.
all right.
Need a pointer.
I'm not sure. So we are in the hunt for rocks, both. You know, yeah. Wow, that is pretty
Brittle.
pretty brittle. That's not really the rock we're looking for, but I kind of want to just grab something and put it in the box, and then we'll keep an eye out for something more substantial later.
Grab what you can better.
This is an indication that this rock's been pretty substantially altered. While it still has some crust on it, the underlying rock has been transformed a bit through interaction with the seawater. The first sample, I'll do the sample entry up here.
Okay.
Hmm?
Hey, just checking in. How's it going?
It's yeah, it's not a great rock, but it's something. So we'll keep an eye out. It's mostly crust. Disintegrates as soon as we touch it. Yeah.
Did you guys get some master? You just went straight there.
Do you want this?
Straight in.
How and did you have to do a lot of water filtering for the CTD? Or
It's still going on.
okay.
Yeah.
I'll check them.
I think she's bottle 7 or 8 right now.
Oh, she's
Yeah.
pretty close. She only has 10. Alright, I'll check them. Did you guys do filter at all or no? Yeah, actually, no.
Yeah.
Great.
Alright, stand by, man. You wanna look at that before we put it in the
Yeah, sure.
alright,
Bring it on in.
bring it in this one. We'll have watch out for it. And to your right. Wow. I think it's super.
Yeah. Let's just go for whatever the biggest chunk you can get.
This is right
There.
below us. We were
Yeah.
trying to pick that up, man.
You can go back for the older stuff too.
This
That
might be easier.
yeah. Whatever's easiest.
Could use
Yeah.
some, handout. Let me use the other arm because it has a little bit more
Alright, go
pressure.
for it.
It's a bit of pressure.
Just try to get the
Yeah.
blades.
Try again.
So, you just rock on it for a little while. Just whatever.
Yeah, I mean, we're not gonna go specifically for rocks. Just if we see something, we'll stop.
I love that. So,
Yeah.
we should do that. I'm a big fan of rocks. I think we should just do rocks like that.
I mean, I think you can never really underestimate a good
Awesome.
rock.
Right there, bud.
Yeah. Just hold that shot for a second. I'll do manual focus here. Is it on manual
Yeah.
focus? Yeah.
There you go.
Okay. Just hold it for a second. And can you rotate it? Or is that gonna probably break it apart?
Because I'm gonna drop it.
All right, yeah. Let's put it in the bucket, rock box.
Barely moving that.
Alright. Report you out then?
Can you zoom out, Steve?
I got you one second.
I can. Yep. I can come up.
Got it.
Got it.
Good being your set. Everybody move it more.
Live box, right?
No, in the rock box, the right one. And that a bit more. That you should be pretty close.
Can you pull Charlie?
Yep. Keep going. There you go.
Steve, I'm just gonna make a quick comment about the
Yep.
quick turnaround and that's.
Pretty good there.
Okay.
Hey guys, just to address some of the questions on Zoom and, sorry, not Zoom, YouTube and Facebook about the quick turnaround. We have 8 ROV team on board so that we can 24-hour dive. So we are going to do a series of 3 dives here at Baker. Plan is to dive this, as many of you watched yesterday, we dove for most of the day, put the vehicle in the water, came out at about 9 o'clock local ship time. So we're currently working in American Samoa ship time. So it is just after 3 a.m. here. And did a CTD dive while the vehicle got turned around, data got copied, and then we were back in the water
This is,
just after
before we go, zoom
midnight.
back in on that rock.
And about a 2.5-hour descent or so, and we were back on the bottom. So the plan is to do the same turnaround after lunch today, and then go shallow again. So there'll be a series of 3 dives here at Baker, which is the plan, as long as weather holds and current holds. So we are set up on the ship for 24-hour operations. So dives will happen at any time
Yeah, I
of
thought I
day
almost
over the
had that
next couple
focus.
of weeks.
Hold on, it's coming back.
So thank you for joining us. Again, ask your questions, reach out to the science team, and come along for the ride.
There we go.
Do you want to try and grab that?
No, I mean, this really isn't the kind of rock we're interested in. It is a rock, but we're interested in something a little bit more solid. But just want to get, can we get lasers on this area?
Got it.
Great. Thanks. All right. Try and get it on the rock surface. All right, great. All right, we can start heading up, and we will keep an eye out for some rocks, since that was not the ideal kind of rock we're interested in for crusts or basalt. There's pretty heavily altered stuff down here. So we're interested in a rock that's pretty rounded. We're not too concerned about having to break it off if we need to. But something that looks crusty and not so angular. I'm going to try and put those comments up because I haven't been able to see what's going on. Let me see.
Move that issue real quick while he's putting it on the way.
There we go. Okay, got it. There's a rat tail fish. Or a mackerel fish. Pretty characteristic of the fish fauna at these depths. Okay. So this is our first kind of our first overnight dive. I mean, haven't stood any 12 to 4 morning watches yet this cruise, so this is a new experience for a lot of us in the control room. We were able to turn around the vehicles pretty quickly, got our samples off, and pop back down for another couple kilometer long transect up the side of the seamount island from roughly about 2,500 meters up to about 1,500 meters today. It's looking a little soft right now, but slope should pick up as we move up. Shrimps. Lots of xenophyophores, these single-celled foraminiferan houses in the sediment. These are these small sediment-colored structures you're seeing.
Great. Do you have a new position?
Right.
Right,
Okay.
it's time for
Mhm.
unless you need something.
No,
right to the
we're all set.
Station. Waypoint. Make sure you're on station, and make sure you're on station. It's the one with the green line over it. So it should be. The leader, yeah. Alright, cool thing. Just get rid of that line. Now you're fine now. The line was going to.
We had a pretty spectacular end of our dive, up at about 330 meters on the other side of the island today. It was 380 meters, sorry. We had some amazing deep water corals, as well as sponges and stylastrids. Basically, a whole community of hard corals growing on the underside of a pretty good-sized ledge that were leaving quite a debris trail down the side of the seamount. But you know, immediately after we indicated we were going to head off bottom, we ended up seeing something that looked pretty unusual, and we ended up in a garden of soft corals, which was pretty striking to see that density. But it looked like it was all one species. Is that up there? It's a sea cucumber. Okay. Is it? Yeah. Sea cucumber.
Yeah,
Okay.
any of these rocks, or-
Yeah. I think these are pretty similar to the type of rocks we were getting. I'm looking for something a little bit more substantial.
Okay.
Although we're seeing some glass sponges as well as some anemones. Yeah, this texture of this rock is pretty similar to what we were seeing down there, and not much relief here right now, but the slope is pretty appreciable. Mostly sedimented though, with occasional pockets of hard substrate. Of course, the hard substrate's usually what we're looking for. The same is so for today's dive, where deep water corals and sponges are often attached to these hard bottom habitats. Although perhaps less so in the past few days. We've seen a number of types of sea pen species living in these soft sediment environments, but it's really probably too deep right now, and it's too difficult of a slope for sea pens to grow in an appreciable abundance. I feel like these rocks would be good if we could break them, and I'm just not sure if it's even possible to break some of
right
these.
There, and it's kind of an edge right there. So it'll be
That doesn't look promising. It's kind of low relief. Anything higher relief that we could break off? We really do want a chunk, like not just like a chipping of rock.
Over to the right there, there's some smaller pieces.
Yeah. Can I tilt up this camera?
It's all your one.
Oh, yeah, there's some corals in the distance as well as some of these rocks. Yeah, maybe. What about these two right here?
Okay.
Those look kind of neat. Looking at it now, I'm not so sure that those are actually movable. It looks like a rock pile over here too. There's a couple of good candidates here. This one also, possibly a good-sized one.
Is that a metric coral or is the
Which one?
coral?
In the distance? I don't know yet. I think we'll go take a look at it after we're done here. I just want to make sure we get our rocks for our colleagues.
Stable. Seeing what you can push around.
Knock them around a bit so that we can see what's loose.
Hold on. Dig within there.
Did that shatter as well? Is it starting to fragment? Either this one or this one. Yeah, I like this one slightly
One
better.
Right in the middle. To the right. Yeah, right
This big one?
there. Yeah.
Yeah. Can you see if that's movable?
Yep.
Yeah. Can we pick that one up? That one looks beautiful.
Can we move for you?
Yeah.
I want to get you closer. Alright.
I know it's big, but it probably would fit perfectly in the box. Before we pick it up, though, can we get in close and image it?
Yeah, I'm gonna jump this a little bit closer.
Sure. Yeah, I like this rock a lot.
Be alright there.
Can I push in an image?
Here you go.
Okay. Do our sample in situ photo. With lasers. Alright, and now we can go ahead and set up to collect. This is going to be sample FK21-248.
Turn a little lower, I can get like the center. Like, yeah, it's not for you. Catching that one, catching that one. Go right there.
Yep.
Alright.
Just hold it for 2 seconds.
That's right there.
Alright, that looks good. We can move to the box.
Alright. Alright, fortune out.
Grab it again. Oh.
For folks unsure who are writing in, it took us about 3 hours to sample all of the biology, mostly biology from this morning's dive. Of course, we have a number of things still going on. We have some experiments that are running live on the ship right now. We also have a number of specimens that are drying coral, for example, are drying in our lab. And we also have, we did a quick pit stop to do a CTD cast nearby where we're diving right now. And that CTD cast brought up some water from as far down as 2,000 meters in some Niskin bottles, carousel. And some of our team members are in the lab right now working on filtering that water primarily for microbes that might be in the water column. So we have been nonstop busy since we got out of the water last, and we will be through the day today.
Yeah, I'm frozen. So you. Alright.
You let go and stand up.
I'm frozen. So. Break that.
We're just picking up a supplementary rock to complement the one we just took. We're looking for a very specific type of rock. As you noticed in the shallows, if you were watching us earlier, the rock type is totally different when we move up above 1,000 meters or so. It turns into this more recent carbonate limestone, basically, that's produced by corals and coral reefs over geological time. But what we're examining and exploring here is a different type of rock and a different type of substrate entirely. We're looking mostly at basalt rock that's covered with an iron manganese crust. So basalt is the rock type that is extruded out in the form of lava from the seafloor when these volcanoes are erupting on the seafloor. So it's a very dense, very hard and heavy rock type compared to the carbonate. And a lot of the coloration you're seeing is that crust from precipitation of iron manganese oxides onto the rock surface over millions and millions of years. That's a good sized rock.
Yeah. Don't crush your baby rock.
That's alright, we can tell it apart. One of those rocks is
Small ones.
yeah, is like Swiss cheese, full of holes, and this one's pretty solid.
Alright.
Perfect size for the box, though.
Spin your jaw. I'll stop. Back up. Alright, spin your jaw counterclockwise. Keep a little bit more. Alright.
Am I hitting anything?
No, keep going counterclockwise. And go out. Just go out. There you go. Just drop it. Drop it. Alright.
Thank you. Perfect.
Break this.
Good. Alright. Right now, you just have lock on
Okay.
and head hold.
We have one person.
Next, yep, next order of business. So we got another rock since the last one was pretty heavily altered, and
Yeah.
this one's really solid. I like this one. We're gonna go check out that coral over there and see what's up.
One on the right?
Yep, and after we take a look at that one, we can proceed up. I honestly can't tell what it is from here.
Have you seen, is it the first coral?
It's the first coral, yeah. We've seen corals and just a couple of black glass sponges in this immediate area.
What we're gonna do is we're gonna come around and look at it from the other side.
Perfect.
Just to get a better view.
Yeah, that's great. I honestly didn't expect to see anything this big, this soon in the dive, but this could be a good omen for some of the substrate type we might be having upslope.
Hey, don't jinx us. Just kidding. No. So far, Baker has been pretty interesting.
Well, surprising, yeah. There's a decent current here. It looks like it's coming straight down the slope.
Oh, so you did get a rock.
Too, yeah. This one was kind of brittle
saw
and
that one.
crumbly. Yeah, this one's solid.
Good.
Is that like a rubble or just like a big sponge?
Is that what?
Is it like coral rubble or
Oh,
just like
that's a good question. I think it's a dead sponge around the side, but I can't tell until we get a little closer. It looks like a dead Walteria sponge bottom behind it for me.
Yeah, yeah. Some
Hard to tell.
rubble. Mm-hmm. I think. Can I push in? All right. Gonna zoom in on this white coral. This actually looks remarkably similar to something we sampled the other day. This is a type of bubblegum coral in the family Paragorgiidae, and kind of gets his name, bubblegum coral. So it kind of looks like wadded up, dried bubblegum. But this coral, not noticing any associates. The polyps are completely sucked in here. If the polyps were out, it would appear more feathery. But I'm not seeing any. I think I saw one polychaete worm closer to the top, but yeah, this is just this colony. Not many associates at all, and but not decently large. Just putting lasers on the colony. We're looking at something like maybe half a meter tall, maybe a little bit more.
That is pretty tall.
Yep.
Especially for being a lone ranger.
But good imagery at that. I think we collected a piece of this at our previous deeper dive, so we're just going to image it and move on upslope
Okay.
on our track. All right. Good to go.
Let's go.
So Steve, I think I'm going to compliment you very publicly. I've read your blog. It's awesome. So for those of you who haven't read it, Steve wrote a beautiful blog. It should be accessible on our cruise webpage, I think. And yeah, it's got some great information about what we did last time in the Phoenix Islands protected area in Kiribati, and how the work that we're doing right now sort of fits into that overall construct of looking both previously mostly south of the equator, but now north. And we're basically just about at the equator right now, which is pretty fun. And Baker Island has been pretty good to us so far, knock on wood.
Mm-hmm.
Our last dive ended right around what?
380.
Yeah, and we left the.
It was a good one. There's a trend happening. I've been noticing the 400 and shallower depth ranges full of surprises. There's some like crazy high densities of things at certain islands at certain depths. The terrain is pretty variable. We were in these kind of karst dissolution features with these crazy overhangs and blocks falling off the wall. Yeah, fossilized corals, pretty interesting stuff. So that would get my vote if we got a chance to come back here one day.
Well, they did say they really did leave us wanting more. We left just at the end at one of these incredible, really thickly and densely populated,
Should we meet an adjustment?
seemingly endless, but we never actually make it to the end.
Yeah.
PRIMNM forest, which was pretty cool.
Yeah, that was high density stuff, and we got a Niskin to capture kind of right in the middle between that plus all the water sample associated with the stony corals we were seeing for our eDNA analysis to try and detect if we can see the the genetic signatures of these corals in the water column based on the mucus they shed.
It'll be pretty interesting to see if your sample is dominated by those scleractinias or dominated by your
I think it's going to be
primnoids.
pretty much everything. Yeah, I wish I had more Niskin bottles.
You would have filled them all up
I
at
would
that
have filled
site.
them all up. Yeah. No, yeah, I need to watch my count because I only have 50 samples for the entire cruise. Well,
50 filters.
50 filters. Yeah. Otherwise, yeah, I actually I saw on Twitter that there was a blog posted, but I didn't know it was mine. So I'll have to go through and maybe I'll do a little Twitter message with the blog kind of describing in more detail some extra bonus features, a little bit more about some of our past findings as well as some of the other analyses we're using to explore deepwater coral and sponge biodiversity in the northern Phoenix Archipelago.
It's a great plan. You can also check out Steve's awesome paper in Frontiers of Marine Science.
Special issue on Pacific deep sea discoveries. A lot of really good stuff in there.
That worked out pretty well. You know, sorry, just going back to the eDNA conversation. Out of all, you pack for these trips, and this one was in particular was really the logistical hassles of packing during COVID and trying to source everything because a lot of things are hard to find. But one thing I never thought we'd have too few of is water filters. We're running out for ours too. Did you know that?
No, I didn't.
Yeah, we had 60, and we're almost done.
Oh, for the CTD or for the
For everything, for all of our water column microbiome work.
Wow.
So we're really almost out. We're
Can,
now rationing filters as well.
So no chance in getting delivery airmail out here then? It's a little expensive.
I mean, we could find one of those companies that say we'll deliver anywhere and challenge them, right? So
Yeah, right.
See what they can do.
Prime doesn't service the islands.
Jeff Bezos,
Pacific
if
Remote
you're listening,
Islands.
just so you know, we're yeah. So let's see what you can do.
Somebody will use it one day. Yeah, but yeah, so we do have to bring everything we need with us, and fortunately we have pretty good on supplies for sampling biology and sampling rocks. Those are pretty easy stuff.
No
rocks,
filter needed.
No filter needed for the rocks. We just put them out to dry a bit, and image them well when they come back up on deck. Very nice relicanthus right here. This is a type of sea anemone.
Somebody in the ROV team, I don't know who it was saying one day there'll be a cruise where everyone's focused exclusively on anemones. It's true. We note them, but there's a big diversity of anemones down here.
Yeah.
And they are really big, a lot of them.
Yeah, relicanthus is really interesting. It's a few years ago, I keep saying a few years ago, but it was more like 2014, there was a paper published that found that relicanthus was actually a completely new group of animals within the anemones. Let me see.
Wow, you pulled that up quickly.
Yeah, I know. We see them quite a bit. They're actually fairly common in Atlantic and in the Pacific.
Is that another arrowworm? That's what it looks like.
Yep, that's acorn worm.
Acorn worm.
Yep.
That's really neat.
Can I
Sorry.
tilt up a little bit to
Sure.
look up? Perfect, right there. So I have pretty good relief here and lots of nice botryoidal crusts. If we could break this off, this would be like our prime rock sample, but usually we have to settle for something that's already fallen off. But if we had a jackhammer underwater, this would be perfect stuff.
We are collaborating with some folks over at the U.S. Geological Survey to pass these rocks over to them to get a little bit of information about the geological history of these seamounts. We know they're about 80 to 90 million years old, but each specific seamount is a little bit different in terms of its formation, its geological history, its seawater chemistry, its crustal accretion, a bunch of different details. So I'm not a geologist, I can't speak to that, but our geological colleagues are going to do a great job of taking all of the, literally going to send them a box of rocks and get some information about this area, which is, it's really, we're in a U.S. marine protected area, so it's really great to be able to collaborate with our colleagues in government to help with that. You were about to say something geologically important.
No, I'm impressed at the relief. I didn't expect to see this so soon, but it's nice crusty relief. Some sponges here, glass sponges, this stalked glass sponge thing. But sponges, yeah, in contrast to our shallower dive, sponges rule the world here. And echinoderms everywhere.
Everywhere.
Everywhere. Voracious deep sea predators. But this is special note for the crust colleagues. Crusty crust.
Oh, highly technical.
I know, yeah. Whenever I see this texture to the rock, it's usually an indication of pretty substantial crust accumulation. But there's rock under there somewhere. But the crust could be several millimeters or even centimeters thick, and in that case, these crusts are laid down on the order of millimeters per millions of years. So one of the ways our collaborators on shore are going to look at these rocks and these crusts. They're going to slice them open with a rock saw and look at these rocks in cross section and see if they can extract out any of the basalts too, because those are useful for age dating these seamounts and islands. Spidodiatomatid urchin, high relief with sponges. Yeah, this is impressive. I'm sure we had some geologists on shore right now. They would be thrilled at this type of terrain. I'm sure corals would be too. I just think that the currents are probably on the slower side to support large corals, but we know that there's that big bubblegum coral down there that they can survive in this type of habitat. I'm just not sure why we don't see more.
Alexis
Jen?
and I were talking about this, and so much of it is probably about larval supply too. So it's pretty interesting to see. You're right. Like this looks like it's perfect for a coral to me, but they may not have found it.
Yep. Yep. Yeah, there's a lot of dynamics going on. Typically, when we are in the shallow waters, we have the highest abundances and densities of seafloor fauna, and as we move deeper into the seafloor benthos, down into the deep sea, past several hundred or thousands of meters, the density of organisms decreases greatly due to the changing quality of the food supply, and not so much the amount of food, but the quality of the food. As you know, a lot of the organisms that live in the deep sea, with the exception of maybe chemosynthetic ecosystems, they derive their energy from surface productivity, sun energy, in the form of plankton that's then sink down and turn into marine snow, which are some of the particles we're seeing all around. And that marine snow is in turn captured by corals and sponges and other planktivorous creatures. But while those particles are sinking, they're also being acted upon by bacteria who are consuming the organic material in the marine snow and respiring, consuming oxygen and giving off CO2. And as they consume organic material, it reduces the carbon quality, amount of carbon available, so that when the particle sinks down to the deepest depths, it's actually highly nutrient poor.
But yet still able to support some life. I remember, I think, it was in a Hedal Troc and Pippo. We went down to about 6,000 meters, and remember there was still some of those large, really large sea cucumbers and a couple of other things. So
Yep.
it's amazing how little nutrients some organisms can survive on.
Yeah, sea cucumbers are fascinating in that respect. They're surviving on just bare, bits of phytodetritus that sink down, and if there was something like a fish carcass or something that drifted down, it'd be like a all-you-can-eat buffet. You'd have all sorts of animals hopping on that energy pretty fast.
It's really interesting. We have a collaboration with the Sea Education Association, and they come to this region every year, well, except for COVID, and they're looking at the surface productivity in the upper 50 meters of the water column, looking at the plankton communities, and it's really interesting to think about those communities and
Yeah,
what we see in
this
the
is great.
bottom. We've just looked a little bit of sediment here and there, not much, but just a taste, if you will, to get a sense of what is on the seafloor now. And it's just these highly productive waters, equatorial waters, right? More so than we ever thought, really. Of course, there's an ephemerality and a temporal nature to it, depending on what's going on with ensostates, etc., but there's a lot to fall. There's a lot of food at the surface, and so there's a lot to kind of feed the deep. Despite the fact that these are relatively nutrient-poor waters when you think about it from a global ocean perspective.
Yeah, there must be something good in the water column. A couple of days ago, we did see a whale shark on our descent. It looked like it was feeding somewhere near the deep scattering layer, which is a layer of animals that plankton that lives down deep and migrates up at nighttime to feed in shallower waters. But that was a pretty cool observation, and I'm sure it's made its way around some large critters there just in time for the next wash.
Sorry.
Gross.
As Steve shoots me with the dirtiest look you can.
No,
while
no,
shooting
no,
me.
no, no. That was not what that was.
You're like, damn it, there's a coral now.
Yeah, we're gonna probably want to take a look at those. There's a few things there. I'm honestly, I'm so tired right now. I'm happy to pass it off.
Steve, what have you been seeing so far?
Rocks.
Okay.
Yeah, this is we saw a bubblegum coral
Okay.
a little bit deeper, but this outcrop's pretty
It's the first one.
pretty awesome.
So much excitement in your voice.
Yeah, I wish I expended all my excitement at the stony coral wall earlier today. So
Yesterday.
yes, I do mean yesterday.
Positions.
Yeah, anything to get a nice view.
Probably from the other side.
I think I'm adjusting the focus while you're flying, so I can. It's looking a little
Oh, just
Yeah,
foggy.
he's gonna put for a little bit. and then I'll
Okay.
Yeah, so we've got a fair amount of diversity here. We've got probably 3 or 4 different species of primnoids, probably across 2 genera, as well as some bamboo corals. I think that one might be a bamboo. The Anthomastus in the upper edge looks like Anthomastus tahinotus, which is a South and Western Pacific Anthomastus species. I would like to look right there
Sure.
at those red blobs.
It's, well,
Sure. And I'm sure you guys are gonna want to poke around in the associates, right? Yeah.
I mean,
There's a few
coral.
things, yeah. Corals associates. I thought these were single polyp Alcyon, but they look like just Anthomastus. Plain old Anthomastus. I am gonna step away while they set up
Well, you
and
can.
hand you over to the next watch. And I will see you in 8 hours.
Good night. Good night, I'm ready. You too. You can go to bed if you want to. Huh? You got it if you want. You can go to bed. You can stay
Okay.
if you want to. I'm just letting you know that you
So we have 2 rocks, that's it.
Okay.
And the next sample is
can say good night.
Do you want us to try and, do you want to look at these small ones with us so you can?
yeah, I'll stick around for a couple seconds. Literally just a couple seconds though.
That's fine. I just want to know if that's something you want to collect.
I am most intrigued by this very sparsely branched Narilla here.
Okay.
I think I know what it is, but it has no associates, so I'm a little indifferent on that.
just try to get position.
Oh, no,
so
yeah. You're
it's-
good.
Don't touch any color.
I was looking down there to see if Debs is down there, but I don't see her.
Alright, time for that, but it's 14:55.
What time do we get on bottom? 13:52.
14:55. We saw high relief coral assemblage. I mean, this. I mean, I don't want to use all the Niskins, but this, on your way
Yeah.
out,
Yeah.
when you see some more on your way out,
Yeah.
and have everything in view, getting
Okay.
Niskin here might be useful.
Cool. I'm gonna look up.
Alright, have fun.
Hopefully that's good enough for you.
Thanks.
Yeah, I think so.
I left my data sheets here. I'm just gonna
Sorry
grab
about that.
these ones.
Yeah,
There you go.
that's totally fine. First, want to just take a look at these. Which look like just kind of, I think like
Regular.
propagating Anthomastus. They
Yeah.
don't look like,
Yeah, I've never seen single polyp like that. It's kind of cool how different life stages.
yeah.
Anthomastus is a mushroom coral. This little pink gummy coral in the background. And normally you see them with kind of a group of polyps. But it looks like we have some new little recruits, so young Anthomastus that probably just propagated off of the other larger colonies. And then we have a Paragorgia and a big Calyptophora, maybe?
Calyptophora? Yeah, I don't know. Here I can get I'll open up our guide. I've been trying to dial in on the primnoids, and some of them are really tricky.
They are. But I think for the most part we've been seeing Calyptophora and Narella. I think in 2017 we really didn't have any idea of what they were. Lots of ophoroids. Little shrimp. We have a bamboo coral over on the right. With all of its polyps retracted.
I mean, I'm no Steve, but I'll call it Calyptophora based on the guide that we're looking at right now.
Yeah. Good enough for me. It's a really good example of the sclerites embedded in the tissue of these polyps. So all those white stripes or slashes inside each polyp is a little piece of calcium carbonate that gives the coral polyp structure. You can't always see them this clearly, so it's actually really nice to be able to see that against that black background. Alright, what else do we have here? How much water filtering is left? Do you know?
Not that much.
Okay.
Yeah.
Is Hannah need help? Or she's already gone?
Brian's been helping her.
Need help?
If you're wide awake, and you want to help them, go through it, but if you have Brian's.
This is definitely a different primnoid. Is this what he said he was interested in when he walked out?
Yes, that is the one. So the sparsely branched primnoid, which would, I believe, be this.
Yeah.
This one.
Trying to get the polyps. Okay, they're all retracting down. Maybe 3 polyps in a whirl, 3 or 4 polyps in a whirl. Is this one we could sample, Russ?
We just did. I'm going to go over it, Brett.
Okay.
But there's also, I wonder if you want to look down.
Oh,
There's
okay.
actually one just
That
right in
looks-
front of the tray there, you see?
Yes, I do. Thank you.
Interesting.
Oh yeah, this might be actually what.
And there's actually one, that's the one to the side,
Ah,
there's one centre as well.
okay.
Yeah, a lot of them.
Which might be even easier to sample.
Okay.
There you go. I can bring the camera out a little bit for you too.
Okay.
Try and get away from that.
Oh yeah, that's helpful.
There you go. Farther.
Great.
Okay, there you go. Should be fairly clear.
Steve, is this the one you were talking about?
No.
It was the other one?
Yeah.
Cup
But
color.
Yeah, cup coral.
so these look like Narella.
But this
This one.
one.
The sparsely branched one is also Narella, but usually it's a species of the Narella with very, very large polyps.
Is it that one?
No, I think it was down to the lower left.
Okay, so it's the one right by the arm.
Yeah.
Okay.
Yeah.
That one.
Yeah.
Okay.
That's usually the sparse branching ones. It's probably something like macrocalyx or hypsocalyx. Can't really tell the difference
Okay.
on the seafloor,
but I think we've already sampled it already. I just really like seeing them because they have the largest
Can you
polyp
move down
size
a
in
little
the
bit?
entire genus Narella.
Just
We-
move the arm out erosion so you can get a better view.
Oh, sorry. Okay.
We've been
Do
working
you want me
with.
to come out? Do you want me to zoom out? Oh,
Working
does
with
that?
that one when we
Okay.
put it in the respirometer, it was so totally beefy.
It was totally what?
Beefy. It went through everything pretty quickly. You could really see the.
Oh, did
Yeah.
you check on the
acceleration of I did.
madrepora?
I mean, they're breathing.
Oh, cool. Okay.
It's hard to look for trends right away.
No, I
because,
know. I just want to know that they're actually
They are.
that something's happening.
Slowly. Very slowly.
Cool.
So,
So I think we're just gonna take a look at this, and then we're not gonna sample it. Oh yeah, there's a lot of polyps on there.
mark which one went in.
Okay. All right. I think we can carry on. You can just come out and kind of look at the rest of the group, whatever you want to call it. So we just had a watch change on our end. My name's Alexis Weinig. I'm a research scientist with the U.S. Geological Survey.
I'm Randy Rogen. I am a faculty member at Boston University, and yeah.
And it is 4 a.m. here for us, so we're your early morning watch for the time being. We are at Baker Island, which is part of the Pacific.
Remote Islands Marine National Monument.
There we go. The PRIMNM. We're on our second dive at this location. Yesterday we did a dive between about 950 meters and 350 meters. We saw some really interesting communities that then turned into a large cliff face of coral at the end of our dive
Face
yesterday.
melting cliff face.
The face smells. So it's been pretty interesting out here. Yesterday we were on the carbonate cap, or the top of of the island, which is made up of carbonate, and today we are down deeper. Can we take a look over here, Russ?
You know, part of the reason we are doing, I know some of you were commenting earlier, why are we doing two dives back to back? And we have this the currents here are really strong. And if there's any wind at all, we have a really hard time maneuvering with such current. And right now we have this perfect weather window where the winds are being favorable. So we are trying to work with that. I wonder if this is Candidella. Not wait, no.
No, it's not. Yeah, I don't think so.
Took a minute.
Looks like just a big colony of Calyptrophora, I think.
Yeah. Maybe this one?
Yeah.
Maybe Calyptra? Or maybe, well, that's just, no, I think I mean, I don't know.
We do our best.
But if I
We're
had to
gonna
guess,
say-
I would try Calyptrophora clarki.
Yeah, I mean, I would just stick with
with
Calyptrophora.
I know, but I'm trying to make guesses, and then Steve can fix it later.
And then we have what I think is some hemicorallium down below too, those really bright pink colonies.
Back to logging. Oof.
Can I just come in a little? Can I zoom in a little bit, Russ?
Yeah, I'm
Okay.
sorry.
Can you see what kind of oofs those are? the top of your head?
That's what I'm trying to.
Keep it safe, but
Yeah, you're good. They're ophiocanthids, so the spiny,
Spiny
arms.
ones. Yep.
But there's some other. I just want to see what these other ones are. Yes. Ophiocanthids, and then we have this precious coral over here. Lots of Anthomastus.
I know.
Most we've seen in one group like that. Okay, I think we're good. I think we can kind of just carry on. See if we can, yeah, find the next patch.
So our team is really interested in characterizing these deep sea coral habitats and sponges as well as their associates. So we're zooming in on all of the critters that are living on and amongst the corals. And I'm particularly interested in coralivores, so looking at the ones who are coral predators, which we've noted them for a long time, but I think when we're systematically characterizing them, trying to understand predation rates and predation ecology on these foundation species in the deep sea. And it is 4 a.m. I'm like rambling.
Nice.
That's the beauty, right? Keep each other awake.
Yeah. Yeah, you kind of got to have two people up here when you're on 24 hours of operations. Keep each other entertained and awake.
Although Baker is doing a pretty good job of keeping us
Yeah.
entertained and awake, Baker
It
Island.
is.
But yes.
Well, yeah. we came off the ground yesterday evening, or off the bottom from our shallow dive around 7:30.
Yeah, right around then.
Which meant the ROV was on deck around 8:30, and we started processing samples. And when we bring everything up to the surface, it depends on how many samples we have for how long it's going to take us to process. And yesterday was about 4 hours of sample processing. So we were finished around 12:30 midnight our time, back in the water. Back in the water about the same time. So it's a quick turnaround for this dive.
We also ran a CTD in between, so it has been 24-hour operations. But again, when you get the right weather window, and when you're in a place that is so interesting, it's hard to leave. And every minute out here is really precious, and so we are trying to make the absolute most of every opportunity we have.
Yeah, and especially like Randy said earlier,
I made an adjustment in your position.
the winds
Bridge.
are laying down, which is a big deal, especially around an island, because we have to make sure that the ship is operating safely and that it isn't going to, make ground on the island. And so having good wind and current conditions is really kind of rare around these features. So we've gotten actually really lucky to be able to do the shallow dives that we have done so far, because it's definitely not always the case. We've been out at these islands in 2017 with the Okeanos Explorer, and then in 2019 with the Nautilus, and both of those had really great dives, but not near as shallow as we got at both of those islands over the last 2 days. My dad said he's watching, so hi, Dad.
Aw.
Which means my dog's probably watching and very confused why she can hear my voice and I'm not there.
Probably still makes her happy to hear your voice.
Yeah.
Yeah.
This looks pretty botryoidal to me, so that really bumpy texture on the rocks. Steve.
Yep. Here, I'll make another comment
Yeah.
on that. Steve's been calling this crusty crust.
Crusty crust. There you go. Yeah, thanks. No, you're good. That's, yeah,
I get that view.
absolutely.
I'm fine with the lookout these days.
It's pretty vertical.
Pretty much.
So,
We're currently at roughly 2,300 meters deep. The temperature down here is 1.9 degrees Celsius, which compared to yesterday when we were in the shallows, we were at about 8.8. So there is quite a bit of difference as you move up the water column, and the temperatures change at a kind of a certain point. You get up to about 500 meters, and it starts to change much more quickly.
and for those of you who want to go back a couple dives, our last one on Howland, we actually were going so shallow that we moved through the thermocline. It was pretty neat to be able to see the waviness to the water. I can't think of a better way to describe it.
Mhm.
It just kind of looks wavy.
Yeah, the temperature change.
Yeah, for sure. The thermocline, yep.
It does.
So, you can't really see those here as we're climbing up because it's much more gradual, but when you're up into the shallows, you really sort of hit literally a water wall where when you pass through it, you can get almost a 10-degree temperature jump.
Yeah, and you see that there are some other places where we're not sure what causes it, but you have really drastic temperature changes in the deep, sometimes flying through other water masses will do that, and you see that shimmer in
Shimmer,
the
that's a good
water.
word for it.
Yeah.
Well, speaking of flying, we should thank and introduce our awesome ROV pilots who are right here with us. I didn't even say good morning to you guys. I just walked in bleary-eyed and sat down. Hi, Russ. Hi, Ben. And we got Debs, our Marine tech, at the back of the room. Hi. Or not in the back, just the other side. Just feels far away. And it takes a lot of people to make all of this happen. So we're here, Alexis and I, but we are not the only ones in the room making all of this happen.
They're making this happen much more than we are.
Correct. We're just chattering.
Yeah.
I wonder if that's the same sparsely branched from NOAA.
I don't think so, no.
It's got sort of thicker habit. Yeah.
Something I like since it's ended.
So, this is pretty hard bottom, pretty good relief. It's clearly the corals that are here are doing really well. They're pretty
Yeah.
big and they're fine. It's just relatively sparse, which is always a really interesting thing to see, to sort of ponder how come there are some here. It's clearly a successful place to be, but are more wonderful.
Yeah, we can take a look at that one that we're coming up upon. And we don't really have many invertebrates on the seafloor either,
No.
which is one thing that I started noticing over the last couple dives, is just how as we get into the more shallow, and by shallow I still mean 500 meters, how many more animals are just on the seafloor and amongst the rocks living in that carbonate versus what is living on this crust down deeper. So I think there is really some truly different communities between the two types of substrate.
And while some of that might be related to depth and pressure, I would bet a lot of that has to do with food supply.
Yes, I agree. Because they all are of.
Check line there.
Okay.
You got these,
And same with fishes. We see a lot more fishes as we get shallower.
I don't see any associates on that.
No, I don't either.
Okay.
Okie dokie. Can we turn the ISO down a bit? Thanks. Yeah, that's much better. So this is a bamboo coral. You can see the nodes, the little brown nodes in between the white skeleton. This is another good example of where you can see those sclerites embedded in the tissue. Randy, you can see some of these polyps closing. See that?
Yeah. No, I mean, I missed that one.
They probably can feel some of the pressure change, the thrust from us approaching, but this
Yeah.
whole row has
There you
essentially
go. I see
closed.
it right on. That's cool. Now, if we can only see them close around a particle.
I know.
But they do move, you know, I've been looking a little bit at their behavior, and they can move pretty quickly. They can close pretty quickly and
Oh,
then
yeah.
reopen pretty quickly, too, which is amazing when you think about the energetic cost of even simple movement down at 2,300 meters deep. It's nice to be able to sort of see some coral behavior. These are animals in the class Anthozoa, so flower animals, aptly named, but they behave and they can move and they can capture food particles in their tentacles and bring them in towards their oral disc or their mouth.
I think that's good. Yeah, oh yeah. Take your
We've been looking for
Take your time.
sort of on the side, looking for different coral behaviors, trying to get a handle on their rates of feeding and rates of movement. One of the experiments we have going on in the lab, geez, I can barely speak, I'm like tripping over my words. One of the experiments we have going on in the lab is looking at basically the metabolism
Yeah.
of these corals using respirometry as a proxy. So just looking at their breathing rate, their consumption of oxygen in the lab afterwards. And we're doing all kinds of interesting things to look at their oxygen consumption under different conditions. Last night we ran our first scleractinian experiment.
Yeah, which I'm really excited about.
So we looked at Madrepora from, let's see, the deepest ones were around 800 meters, maybe 900
yeah,
meters.
yeah.
And then the shallowest ones were from around just around 400 meters, maybe a little bit shallower, maybe a little deeper. And we're just taking a look at their basal respirometry to get a sense of what their breathing rate is, whether or not it differs over that depth range. Keeping in mind that Madrepora doesn't live deep. Right around 1,000 meters is sort of where you start to see it. I don't know, that might be an incorrect estimate.
It's 1,200, but not in the larger aggregations like we were seeing. Yeah, the respirometry setup that Randy brought out is really cool. It's a very small, so I've run a decent amount of experiments with the cold water coral known as Lophilia pertusa, which is kind of the lab rat of the deep sea, if you will, because we can keep it alive in labs, but we don't have Lophilia out this far in the Pacific. And most of those experiments required taking live coral back to the lab after collecting it. And what we're able to do out here with Randy's setup is actually run the experiments on board, which is really helpful because these corals would not stay alive until we got them back to Boston. So it allows us to run experiments in real time when the corals are fresh, and it's a very transportable little setup
unit
that she has. Yeah, it's very small chambers, but it works really well for these corals that have the smaller polyps, which is luckily what we see out here.
Yeah, can't do anything large-bodied. An
Yeah.
anemone, for example, would never fit into it.
Yeah, and Lophilia would not fit into this chamber, but it's cool to be able to do it with Madrepora, which has really small polyps on the tips.
So last night we were a little slower as we were trying something different, but Anna and I have this pretty much optimized now, where within about 20 minutes of them coming up from, or getting into the lab, they're into the experiment,
Yeah.
which is pretty great. I'm really happy to be able to pull that off. Is that a little bamboo in the bottom?
Yes,
Barely
it is.
see it.
Yeah.
So we are logging what we see to give ourselves little guideposts to go back to.
Yeah.
It also keeps me on my toes a little.
Mm-hmm.
sponges,
Sponge, yeah.
sumac, and anemone. Hit the wrong button. Where is it? Here it
So
is.
there's some chatter in the chat about why these corals are the colors that they are, and that's still a pretty active question in the research community. Let's take a look at this coral. It's definitely different than what we've seen so far. Sounds good. And some of there is definitely the possibility that it's just a trait that's left over that isn't actually benefiting the coral in any way, but at the same time, pigments also can have a lot of properties associated with them. So there are some theories that it's protective in some chemical way, but it's just not something that's been tested heavily. This looks like an Acanthogorgia, a really big one actually.
It is huge.
But let's take a look, because with how big it is, I also don't wanna. It might be something else.
We still not have any Acanthogorgia button.
Oh, sorry. It's
Right.
because I haven't been making buttons. I need to make a note to myself to do that when I get off.
I should tell you some more about the work that's going on by my co-mentored postdoc, Stephanie Kennedy, who's beginning to think about different pigments and how it might be related to
Yeah, please.
coral immune systems.
Mm-hmm.
So one of the focal points of this cruise is looking at coral immunity and trying to understand what their immune systems look like. They have pretty primitive immune systems, so do sponges, but we probably share some features already with them in terms of how we respond to different microbes in the water column or on the benthic community, or when they get grazed by a coralivore or a spongivore, when they have an open wound, they can get colonized by bacteria, and so having an effective and active immune system is really important. These are pretty ancient immune systems, and so trying to understand the origins of innate immunity is one of the things that we're doing on this cruise with Anna Gauthier
Okay, should we get
Okay.
and John Keegan and me.
camera for yourself?
Yeah, so this looks like pretty classic Acanthogorgia. Has those really dense polyps along the axis. You saw the one that we brought up yesterday,
I
Randy?
did.
Yeah.
It's so spiky.
Right. They have a lot of sclerites embedded in the tissue, and when the polyps close, there's kind of like a crown of thorns, if you will, around the mouth of each polyp.
So
Here's a
they're
little
astrocytes.
aplaque.
Oh, yeah. And then,
Aplaque
are
offerin.
there any active predation that you see
Actually, this might be. Looks like he might have a polyp in his mouth.
in his mouth? Where?
Yeah.
Oh my goodness.
Yeah.
See if we can get a.
Might try it. We're at max zoom right now, but
So I'm trying to move the camera.
Okay, yeah.
So
yeah, gives us something.
Adjustments.
So we're also looking at, Randy is really interested in coral predation or corallivery, and we've seen a couple times these little small aplaque offerin worms that wrap around these corals, and you don't always see them feeding, but it does seem like it has one of the polyps, or at least is making contact with one of the polyps.
I think we should probably sample.
You wanna
it so
sample
we can
that?
do
Okay.
some stable
Yeah.
isotope work with it afterwards.
And you can see right around where the worm is wrapped around, all those polyps are retracted. So it's kind of, these corals are very sensitive to any disturbance, and they'll pull those polyps in to protect themselves.
If it's eating, it's just starting.
Yeah, that's true.
There's not much
There's not.
denuded tissue.
And we don't really see predation.
There's another one over here.
Okay,
Actually, can we
yeah,
take a look at that
yeah.
before we sample?
So it's
what's
gonna
going on.
scan the coral and see if there was anything else.
Do you see it?
Yes, I did. Or I did. Now I don't. Where'd
It's
it
right
go?
here.
Oh, thank you.
You're welcome.
And it might be that, you know, these corals, once they close up their polyps, they might be somewhat inaccessible to the aplacophorans. So we might be seeing an attempt at predation that isn't super effective, especially because these do have a lot of those spiny structures in them, which would make it difficult to actually get to the tissue.
Although I have to say, it looks like there's another
Yeah.
attempt at sort of
Direct.
direct.
Yeah, trying to get into the polyp.
But you know, it's pretty high up, so if it was just trying and not succeeding at all, I think it might. How mucousy are these? I didn't.
They're
see
not
yesterday.
that mucousy. At least not immediately.
Well, I'm already just amazed that this thing isn't deterred by the barbed wire spikes.
Yeah,
On the,
yeah.
yeah. Let's
Let's see if we
whichever
have.
one is the easiest one to sample. It takes the least amount of tissue. Is
Okay.
probably what we should.
I
go
think
for.
that one up top is
Yeah.
probably.
Do you see another one to the right?
Oh, yep. There's another one.
Yeah. Maybe it's that one that's
That
the
would be
closest.
That was-
superb to get access.
Okay.
That's good.
Superb.
Can we take a close look at it though first?
Yes.
Yeah. See the one in there? Yeah. That eventually is what we'll go for.
The one with
The bottom.
I don't know. I don't see anything active there, but we can't see the other side.
Yeah.
And it looks like it's heading up towards the polyp slowly.
Yeah. I mean, you're right. They are on there, but none of the polyps. There's
Mhm.
not any degraded tissue like we
No.
see when we find sea star predation, or even on some of the other aplacophorans we've seen have been causing more destruction.
All right, which makes this a great candidate for
I think
stable
this is a good
isotopes.
sample if that
Right.
works for you.
So the question is, are they just?
Yes.
Alexis, did I miss it, or did you zoom in on the side that does look like they're polyps or not? On the left?
First things first, Ben.
No, I know it.
Oh,
Oh, yeah,
okay.
that's
See over
good.
there.
That's a good.
I
We
just
will.
didn't, I wasn't sure if I missed it, I was like,
No, you're right. We didn't, but we will go look at that
I
after.
didn't even see that.
Yeah, thanks, Deb. We need a third pair of eyes, a fifth pair of eyes.
Well, it is my morning, it's your, like,
Who knows
wherever.
what?
There's that.
Yeah.
Camera's
All
all
right.
about.
On the cup of
Yeah,
coffee.
she's right. It's right over there
Yeah,
on the side.
yeah.
Didn't even see it. I was so busy scanning for
I know, I know.
aplacophorans, like for worm-like shapes, that I just didn't even. Sometimes search image is a funny thing. Sometimes it can work for you. Sometimes it can work against you.
That is a worm, right? That you're, is it a worm?
It's a mollusk, actually. So it's a mollusk without a shell.
Not as
Um,
pretty as a nudibranch, but
Yes. They're very interesting little from an evolutionary perspective, but we see them pretty often in the deep sea on these corals, and they're I don't know too much about their occurrence in shallow water personally.
Oh, yeah, I see this one.
This is going to be 249.
You want the worm, right?
We do, yes. So just a snip of the coral with the worm. It doesn't need to be a whole lot of coral.
Okay. I got the one right behind you.
I see it. Looks great.
Yes, the things with label-based business
Yeah, somebody asked what's the size of the colony. This is pretty big, especially for Acanthogorgia. We have lasers on when we're zoomed out, and those are 10 centimeters, and it looks like this colony is at least 50 centimeters high, which is quite large for. So you have some stuck in the top of the jaw. Nice.
that's perfect.
Yeah, back in the camera.
Did you catch which quiver it's going into?
We don't have anything in any quivers.
I was gonna do 16 if that was alright, just
perfect.
it's easier for us to
Yep.
get
You
access.
want to take a photo of it first?
Yeah,
There you go.
I got.
Right. There you go, camera's yours.
1 second.
Let me just.
Good.
You're good.
So,
Okay,
hey, we can
we're
zoom in
good.
if you want, but
No, we're good.
okay. Got it when it was on
Yeah.
the seafloor.
Set.
Nice job.
Great job. So we had a question about who's controlling the metal arm, and that is one of the ROV pilots. Ben is on the arm right now, so they have essentially little miniature arms in the control van that they can move around in order to move the arm around underwater. So it's actually a very difficult thing to do, but all of our pilots are very good at it here, which makes our jobs a lot easier too. They're very efficient at what they do, and so that means that we have more time looking around the seafloor, because it takes a very short amount of time to sample normally, which has been good.
It's
I think.
amazing,
Oh,
yeah.
actually, we want to look. Sorry, Russ.
That's okay.
At
We're
the-
gonna look at that area on the left real quick.
Do you want me to come up and reposition?
No, I think you're okay.
Alright, let me just push it on first then.
Sounds good.
When I first started working in the deep sea, and I'm still kind of I still
Great.
spend more time in the water than on the water. You know, I thought I would really miss my hands, right? Because when you're in the water, you have total agency on your own. Whereas here, it's sort of collaborative agency where you're working with a team of people to try to get to what you need. But I have to say that the pilots here are so good that it really does feel like you have your hands, and the collaborative effort is really nice.
So there is some degradation. So this slough,
sure.
this is like sclerites left, but no mesochyme, right? And like these are like essentially dead polyps.
That's true, but we haven't seen that really too much.
Yeah, I don't know how much we've been looking at Acanthogorgia for that.
It's true. I also don't know how much. I don't know that we can attribute this to
No.
cofference. There's nobody around, and we don't see the same kind of damage where they are. But what I wouldn't give for a 2-year time-lapse camera.
What?
What I wouldn't give for a 2-year time-lapse camera.
Oh, I know. You can answer a lot of questions in the deep sea with more temporal experiments. It's just hard to do that. All right, that's great, Russ. Thanks. Sorry, I thought somebody was back there. They're not. Everybody's sleeping.
And everybody's sleeping. Is there something you need?
Nope.
I'm logging.
Yeah, you're doing great.
Well, thanks. I'm just trying to stay awake.
I know.
I'm not
I'm
caffeinated.
feeling more awake now. I've been up for almost an hour.
I'm trying to engage you a little.
Huh?
I'm trying to play with,
Yeah.
but I have not had coffee deliberately because I don't know,
When do you need to go to
Wait,
sleep
wait.
again?
which is kind of a funny situation to be uncaffeinated. But if I have coffee, that's it. I'm done for the next 10 hours.
Oh yeah, it's a long cut.
That's a long time. Was that a bamboo that we saw standing up? I forgot to mark it. I think it
I
was.
don't. I'm not sure.
I'm just gonna list that. These look like they could be. I can't tell at all because they're flat onto them.
Yeah. See,
I can't even tell if they're
Sounds
pink or
good.
white. They look pink.
I think they're
Yeah, they could just be the yolk.
They are so plainer though. Look how thin.
Exactly. Barrett, where'd you at?
Or a Corallidae. I just don't know yet. The ones we brought up yesterday, we sampled a hemicorallium that had all those flatworms embedded in the,
Oh,
or polychaetes, not flatworms. that burrow, and this is looks like hemicorallium to me. Had both of them have those astroschema brittle stars.
Yep. There we go.
Yeah.
And there's an anemone. Zoanthids?
I think so.
I can't. You'll have to zoom a little more. It looks more like zoanthids now with it.
It's just the having multiple of them is what makes me think.
Sometimes you have multiple anemones right next to each other that aren't connected, right? I think it's a zoanthid.
So this is a precious coral, the common name for that group. Both of these, the one in the background also. That's great. Thanks, Russ. And these are in the family Corallidae, which is all the precious corals, and we see this hemicorallium pretty regularly. We saw it also much shallower yesterday. It's interesting to note that distributions of certain corals can be quite different. Some are really only found, in maybe a 500 meter range, and others are found across a 2,000 meter range. There's kind of sweet spots for certain distributions, it seems like, but we're still just getting a handle on some of that because, again, we only get to have glimpses of this space every time we bring an ROV down. We're just doing a track line up a seamount, which is, like Randy has said, shining a flashlight up a mountain in the middle of the night.
On a teeny tiny little hiking trail.
Yeah.
Well, that's actually Jan Whitting who said that, and I just loved it so much I keep repeating it, so I'll give credit where credit's due here. If we see sponges, we should zoom on them too. So
Okay.
we're looking for spongivory also, and I feel like we're so focused on the corals because they're so charismatic, but little sponge love,
Yeah.
especially since it's so sparse here. It's pretty interesting.
Really rocky terrain.
Totally. And yet,
Kind of reminds me of some of the landscape in like Lord of the Rings, where they filmed in New Zealand, and it's all.
oh yeah, definitely. I was actually just thinking about my kids. We one of our favorite places to go to on vacation is to Arizona, and half the reason is so they can hike, but what they call it rock scrambling.
Yeah. Yeah.
Because it really is when you're in some
Yeah.
of these formations in Arizona, and you're just clambering up these. And if this weren't 2,300 meters and 1.9 degrees C, and they would love it here too.
I
And
don't know,
underwater.
it's pretty vertical, so they'd have to be little billy goats.
Cucumber?
Yep.
I think so.
Holothurian.
Yeah, he's trying, we have that cliff there.
It's like stepping stones. Doom, doom.
It's funny to try to transpose this into a terrestrial landscape and imagine what it would
Yeah.
look like. Yeah.
And somebody had asked or mentioned earlier, is it igneous rock, and underneath the crust would probably be igneous rock. So these are features that have been formed by volcanism way, way, way in the past, and over time this manganese crust forms on top of that basalt or that igneous rock. And like we talked about earlier, we have at the top of all of this, when you get more shallow, there's carbonate on the top, because at some point there would have been a coral reef associated with the top of the seamount. And so that carbonate is left over after that coral reef has died.
We were looking at some ancient fossils and fossil corals yesterday. I don't know how old they were, but probably hundreds of thousands of years, and it was pretty awesome.
See another little cucumber down there.
Got it? Thank you. Still busy annotating our previous cucumber, which seems to be the dominant non-coral associated invert.
What side of the island are we on? Do you know?
Yes,
Or is it in the?
I'm gonna orient it in my head in a minute.
Yeah. I just don't even. I didn't see the track yesterday, so.
We are at the southeast edge. Yes.
What's up, Deb's dad?
Southeast. Okay.
Unless
Huh?
Friends on there.
Oh.
Chief.
you have a friend on there.
I think. Yeah.
That's awesome.
Oh, can we take a look at these little white?
Yes.
Just not sure what those are.
Little cenophibers.
Or little sponges? I'm not sure. It looks.
Let's get a little closer.
I'm not sure either.
They look kind of calcareous, don't they?
They sure do. Which would.
It's okay. I'll come out.
Whoa.
So I'm getting closer.
Yeah.
Getting closer to it as well. Set up at 80.
Well, if it is a xenophinophore, I can't even say that.
Xenophyophore.
Let me go back to logging. It's a cute one. They are
Okay. Let's see.
Okay.
pretty cool. They're foraminiferans. They're single-celled organisms, but they have multiple nuclei. And don't ask me how exactly they form, because I can't tell you.
I
Maybe
know.
you do.
I don't know. That does actually look what it is. That looks like
So
agglutinated sediment. It's pretty much what a xenophyophore is.
Do we have an other button?
We should.
We don't. I guess this is as close as we'll get. All right.
Yeah, it's definitely sediment.
Yeah. And then so they get this sediment that kind of accumulates all around the outside of them. But they're amazingly structured for being such incredibly simple organisms.
Yeah. They're always very intricate. This one has like kind of that fan, but sometimes they're little tubes and tunnels, and they're hard to collect.
You know, they really look a lot like
All right, that's good.
a really shallow water algal species that you find amidst the seagrasses that we see all the time. It's really almost exactly the same shape,
Oh.
which is obviously
That
just
or
yeah,
oh, okay.
which is obviously just convergent
Oh.
evolution. What is that?
Anthropogenic. Looks like a
So
bottle.
we gotta take a look at it and document it.
We take a look at that green bottle right up there.
I'm sorry to say we've seen quite a bit of litter, not a ton, but
Not
given
a
the
ton.
small tracks that we're running,
Yeah.
we've seen it on just about every dive since we've been here.
But this is, I mean, there have been inhabitants on this island
For
before.
sure. But and this is, it looks like it's probably glass, which as far as impact goes is pretty light. I mean, really, it's just sand.
Yeah, once it
Melted
breaks
sand.
down.
Yeah.
Yeah.
Okay.
Yeah, if you have to choose between plastic and glass, I always say, from an environmental standpoint, glass is much better. Well, actually, I don't know what that is.
Yeah, me neither. It's definitely-
That's not a bottle.
Oh, it's a shell.
Yep.
Oh.
Uh-huh. So there was some military action here way back when, and we don't have an option for that.
No, I'm sorry.
That's all right. I'm just going to use litter and write artillery shell.
Yeah.
Yeah, it looks like
Can you tell me anything about it? I don't know very much about these things.
it's a large round. I'm not familiar with that sort of style rounds. 30 mil maybe?
Any idea?
Back out a little bit.
50 mil?
Back out.
Back out.
Yeah, just to get the.
Lasers.
Yeah.
Let me bring them up.
Okay, so it's about
It's
It's 100 long.
that, yeah.
yeah.
100 mil long, so.
So yeah, 30 mil.
Yeah.
So that's pretty interesting. I've never come across an artillery shell before.
It's very
Oh.
well decomposed.
It's been a very long time.
Yeah.
Well,
Yeah.
right. So there was occupation here in the during World War II.
Yep. A lot of these islands were sort of strategic military
Yeah.
positions during World War II. And a lot of battles were fought on islands not too far away. The Battle of Tarawa, for example, which is in Kiribati, and it's 1,000 miles away, but it's sort of reminiscent of these kinds of places, sort of a small, low-lying atoll.
Will you grab it?
All right.
Okay.
I mean, like, as long as you're sure that it's.
So we're gonna pick up some trash here, so we don't leave it on the seafloor. It just kills us to think about
It's probably.
leaving this here in the deep sea. It's our problem. We can pick it up and get dispose of it properly. Yeah, in the rock
I'm
box.
going to porch it a little bit for you.
What?
There's a camera in.
So this is going to be sample number 250.
Mm-hmm.
You got one set for me? Alright.
But it might be something where it's like, I don't know if there's any more room on my thing, but sometimes trash is hard to pick up.
Yeah, sometimes like there was a bag we saw earlier that was so filled with sediment, there was no way
Oh.
we could have picked it up. I think so. But whenever we see seafloor, we're definitely trying to clean it up everywhere we can.
Oh, yeah. If you were to go to places like Seattle, there's just full and stuff like that. They would come back into the sound and just dump ordnance over the side.
Oh.
Years.
Very low, San Diego too.
Brian was telling me about some time on the EX when they thought they were going to see this like incredible, they just saw this structure off in the distance, and it turned out to be a bunch of washing machines that had come off,
Yeah.
tipped over.
I
I
think
don't
they
think-
even saw it. I think they found it in the multi-beam, so it
Oh.
looked like it was the shape of what would be a ship. And then
Oh yeah.
when they went down, it was a container of washing machines.
So we published a paper on marine debris from this area, actually, which was I don't know if you've seen that. It's pretty cool. We put it in that special issue, but what's nice about these protected areas is that comparatively speaking to some of the more coastal habitats, there's relatively little marine debris. But around these islands, where people have actually used them, there's always more than there is, obviously, when there are places which are truly uninhabited or have not ever been inhabited. So it's amazing that even though these islands, I mean, if you were to see this, you'd laugh. Who would possibly live there? There's not a single tree. It's, less than a mile long, or just about a mile long. There's nothing. It's all seabirds. But it's, people were here for a little while. I think there were 4 people outposted here for a bit. And anytime there's people, there's impact.
We don't need. We can keep
And
going.
in the deep sea, it's lasting impact. We see it for a long time. It doesn't just disappear. It disappears from view from the surface, but not down here. Why can I not find urchins? I must be staring right at it. Here it is. It's right in front of me.
Speed up with this stuff.
Yeah. There's a big xenophile. 10 centimeters.
Oh, there it is. Second to last bullet, Deb. I don't think it's.
I think it's
Good one is.
Found which lights.
Yeah, we got plates last time.
Do you need license? What do you got for?
No, no. Like a full crate we left on the seabed.
Huh?
Somebody threw it
Were
over
they
the side.
Hanging up
Were
by the
they
wall.
broken?
No, no. They were all nice.
Wow.
It's like they hadn't been used. They just.
Where in the world was that?
Come off of a ship or some sort. It was off the coast of California.
1958 or something.
Oh, wow.
Randall,
Yeah.
I updated this, but if there's something wrong, just feel free to change it. Based on what they were saying of recovery, like this is the next one, the shallow.
Yes.
Oh, okay,
here,
yeah,
and then
okay.
deployment, and then, but I don't know what that. The turnaround time is gonna depend on the video trend.
I think there's a, Deb, correct me if I'm wrong, but I think there's a cluster of 3, like 2 together and 1 a little further apart.
I think there's just 2 next to each other,
Okay.
but one looks kinda like
Yeah.
attached.
And then I think there's another cement on the way too, so I don't know, we'll have to take a look at it. It's been a couple days now since I've looked at it.
This one, unknown 3, is going straight to the far one, and then hitting the other one on the way back.
Exactly. Perfect.
So,
So,
8 hours is just for the far one.
right. So Deb and I are chatting about our next steps, because we're gonna do another dive on Baker after this one, and then
What's our projected? What's our depth for the next one?
So we have to chat about that. We can
Okay.
either go 150 to as shallow as possible, which was my plan, which I think
Okay.
we'll still do during daylight. Then if we want to go back to that PRIMNM forest, which I think we do, we might plan a fourth. Cause it's a different, because you can't go, we can't work through the current. So we either have to
Okay,
go
okay.
4 up to 200 or 150 to shallow, but we can't work 200 to 150.
Gotcha.
Weeks.
Or one. Okay.
The-
I hear you.
yeah, we can't work through. We have to work below or above that current.
Okay.
So we will.
Otherwise it'd be super fun. On that.
Nope. So I think what we'll do actually is, let me turn this on.
Sand.
Back to regularly scheduled sand. Yeah.
Okay, you wanna talk about where we are? I'll put up Google Earth.
Sure.
2.5 years, 2.8 years
Oh
old.
wait, that's not Google Earth, stand by one.
Yeah. I'll, open up a little bit.
Okay, somewhere in the middle of the Pacific Ocean, here's America.
Wait, can people see this?
Yep,
Oh, okay.
here's America.
Well, so.
To
Well,
orient
actually,
you.
it's funny, Debbie. I just called you Debbie. I'm sorry.
Orientation.
It just came out. I apologize. Debs just put up this map of the globe, and she started off with saying, "Here's America," but where she's going to take us to is actually still
Yeah.
America.
In America, here's America. And over here, here's
She's taking
America.
away from me. She's like, "You called me Debbie. You're out."
Randy's kicked off because she called me my I'm just joking. Yeah, here's Howland and Baker Islands. So today's dive sites up here. Okay, go Randy.
No, it's all good. You.
No.
This is your map.
No,
Own it.
it's just my map. It's about 20 seconds of mapping.
Yeah, but filling in gaps is really important.
I woke up just over here when we were doing a CTD and happened to push record. So we got this little bit of the map
Maybe a little
where we're
segment
diving.
here.
Yeah, I've been sitting looking at it for a while.
Great. Yeah, let them. Sounds good.
But we're up at the North Ridge on Baker Island.
So, as Debs is showing you, well, we're about to take a look at a sea pen here, but we are truly in the equatorial Pacific. We are just above the equator. We'll probably cross in the next couple days, which is always fun. But this seamount, this island that we're looking at, is
like
certainly
that clear.
influenced by equatorial currents and countercurrents that are right in that zone, which is influencing the productivity above us. So we're in a pretty oligotrophic or nutrient-poor part of the Pacific, but it's thought that these equatorial currents actually give us sort of a productivity bump right in this zone. And it's a really important food source for all of these corals, including this sweet little sea pen.
Yeah.
Not pretty.
Can we bring the ISO down a little bit?
I don't know if it will go down with the machine.
that works too.
Yeah, there you go.
Oh,
It's not too great.
It's in the
no,
bottom.
that's good.
It's
Thanks.
got little
Yeah. I was gonna go down and take a look.
What is that?
Scale
Some kind of.
worm?
I don't know.
Swim.
Worm or is it a
amphipod?
crustacean? It looks kind of crustacean-y
Yeah,
to me.
yeah. It's either an amphipod or
Yeah. Let me tag it as an associate
Archie.
here.
so cool. The stalk of this sea pin is really interesting. Large polyps all extended out. So sea pins are a type of octocoral, but they have a peduncle or something that is buried in the sand to station themselves versus having a holdfast, which is how most of the other octocorals that we see are attached to rocks. So they're able to occupy this open expanse of sandy area without, without floating away or getting covered in sediment. How are we with this shit? We still waiting?
Just give us another minute
Okay.
or two.
You can, Randy, you can zoom on sand.
It's, well, you know it's one of my favorite things to do. Let's do it.
Xenos right ahead, right?
There is a Xeno right in front of us.
Let's do on the sand too.
Those bush goers are taking amazing samples.
Yeah, it's kind of the only way to do it, honestly.
I love looking at this. You can see sort of the pteropod and the foram tests and pteropod shells, I should say, and foram tests, and looks like some urchin spines and various flock left over from marine snow, and then of course another xenophyophore, which this one is a completely different structure and shape, which is really cool. So again, these are a type of foraminiferan. I don't really know very much about them.
I don't either, actually.
They're multinucleated, unicellular, cryptic. No, not really that cryptic. They pretty much stand out, but they match the color and the texture of the substrate around them because they literally have all of these sand particles attached to them.
And some people do collect them and study them, and I think they often find smaller, like little amphipods and other invertebrates living inside. So, another instance of providing habitat for other little animals to hide in when you're up on this big, open, flat space.
Plains. Let's just call it the plains.
We're just gonna go in.
Not the abyssal plains in this case, just the
No.
seamount plains. There you go, there's a pteropod shell.
Yeah.
What are those dark spots? I always think about
Okay.
the decomposition of photosynthetic material and whether or not you sort of see anything
Good
that
to
even
move.
resembles algal structure, because it does fall from the surface, but here probably not. There's not that much stuff to begin with at the top. This is probably just all sort of marine snow and flock that's kind of come together. So even though it looks sometimes a little algal-like, it's really not what it is. There's certainly no photosynthesis down here, and there isn't enough productivity on the island for even to see what I call deep sea botany, which is sometimes you see leftover sticks or leaves or things that have fallen all the way down, but there's really nothing on that.
Yeah, you
There's
just
no
sometimes
scrub.
see like full palm fronds and stuff.
Yeah, but no deep sea
Or
botany here.
sargassum and other places.
Oh, no, I guess there could be sargassum here too, but I doubt it. It doesn't look like that at all.
Oh, I
Yeah.
haven't seen any. I just know
No,
that
exactly.
I've seen it in the Gulf of Mexico.
Yeah. God, we were just in Belize a year ago, 2 years ago. I'm losing track of time with COVID.
Yeah.
And there was just the biggest sargassum bloom I had ever seen. It was washing up in the beaches in like meters thick. It was
my
been rotting
gosh.
on the beaches. And
Oh.
there was no place to put it except to try to dry it out. And I mean, it was just is littered with microplastics and macroplastics and
But also
debris
probably
and-
bunches of little
yeah,
crabs and
all kinds of other stuff too.
other things.
Yeah, but all decomposing on the beach
Yeah,
is
yeah.
not the nicest situation.
Probably very smelly.
Yeah. So we take undergraduates out there every year for field courses, and it's snorkel field courses.
Yeah.
Again, in the water versus on the water. And usually we see some sargassum, but I had never seen anything quite like this. This was just the piles of it. You could hide and make forts out of it if you could stand the stench.
Yeah.
We didn't, but
Yeah.
you could. What a transition from that incredible rocky terrain
I
we were just
know.
on.
What's the slope here? It's still pretty.
It's not very steep.
Yeah.
If you look in that camera,
Yeah.
it's definitely.
It's interesting that it's so blanketed with sediment, with how steep this actually is. Hey Deb, can you bring up that plan of the day again, just so I can look at it? I just don't remember the times.
They're all subject to change, of course.
Well, right. I just
Yeah.
okay.
Can you pick up?
Right.
First, I just try and keep it there at 75.
We're gonna try to recover right after lunch.
Okay. And then we would deploy again, possibly. Okay.
I'm pretty sure we will.
Okay.
But
Okay.
the question is whether or not we do the super shallow above 150, which I think we should, again, while we have this
Right.
perfect
Right.
window.
Okay.
And then it's just a question. The only thing that would change is line 140, whether or not we transit
Gotcha.
or whether or not we stay and do one more and try to put ourselves back on the PRIMNM forest,
Okay.
or whether we try to go west and then come back, which is not out of the way because we're going to Taitav
Right.
after we go west. And
Right.
this is it's a 3-hour diversion. It's not far.
I mean, that might be a more manageable plan, depending on.
It might be, if as long as we think the weather window will hold.
Right.
If the winds kick up, then there's no point
Right.
in coming back. We won't be able to make it. So it's just a question of how badly we.
What's today? Friday.
I don't know. I think it's Friday.
It's Friday.
It's later today.
I remember Brian saying it did look calm at least as of yesterday or 2 days. Wednesday.
Right through Sunday.
Yeah.
But if we go west, we won't be back here before
Okay.
Sunday.
Okay.
So that's what I just need to chat with the captain about and figure out what works for us. Or maybe we just leave it and try to come back on another trip. But it would be really nice to see what we're here.
Yeah.
It's right here.
Well, and it would be interesting to continue that.
I agree. It's
Track,
a very interesting
yeah.
track.
The forest is,
We
is
didn't have a
cool,
c-
but it did also look pretty homogenous. Whereas if we kept going, it would be interesting to see what else.
Yeah, I
Pretty
agree.
gnarly.
We had a pretty full basket and we had a lot
Oh,
to
yeah.
do, so it was time to pull.
Yeah, I'm not
Oh, okay.
advocating for
Oh,
having
okay.
stayed. Believe me.
Okay.
Yeah.
So I felt pretty bad about pulling us out, but it was amazing. But it was time to go.
Yep.
It's always this dance between what you want to do, what you have to do, what you can do.
Tide, yeah.
Yeah. Time is a zero-sum game out here. Every minute of it is precious, and so we
Yeah, down
do
here
the
it's
best we
fine.
can
There's
to make the most of every minute.
pretty strong currents on the way down, but once we get down to the seabed, it's been pretty good. So there you go.
Okay, I see. So if we do the 150 meters, it's gonna be a pretty short dive.
Yeah.
So we don't have
No, we don't have
very
a long
far
way to go. And we have to be out by sundown.
Okay.
It's very shallow.
Okay.
So we'll try that 1300 is the 1500 is, I hope, an overestimate. I'd like to get in earlier if I can.
Okay.
But they have to download the data from this dive.
Right. It
before they
takes
can
yeah.
turn
The
it over.
turnaround is. There's some shrimp swimming along.
This looks like we might see some structure up ahead a little bit. I can't really tell. Or maybe not. Trick of the light. It's like you than being in a desert thinking you see an oasis.
Yeah.
Shimmer. So, lest you think sand is boring, we are trying to traverse it because our goal and focus is to think about corals and sponges, but this is not boring, nor is it useless. This is an incredibly productive environment. There's a whole bunch of myofauna that live inside of here, as well as a bunch of small things on the surface that we're just sort of glancing over, in addition to the xenophyophores, which you can kind of see poking up. But what this really is, and I think about this all the time, is when people talk about the importance of the deep sea and how they sequester carbon. This is part of that story. You know, this is part of the carbonate shells of surface organisms that have fallen and are now just going to stay drifted gently to the seafloor, where they will stay here and and not really come up for a long time. Although upwelling will bring them up eventually to places where we need them. But this will be stored here for I'm trying to put my finger on the number, but I can't right now. I'm too tired, but
That's fine.
it's stored for long enough that it stays out of the surface oceans, and it's able to transfer that atmospheric carbon to the oceans because all of the surface photosynthetic organisms are able to use some of that atmospheric carbon, transfer it into their bodies by incorporating it into making shells and making other calcareous parts. I'm pretty tired. And then as they die, or as they get consumed or eaten, or as they produce fecal matter, a lot of that just transitions down to the seafloor, where this is what we're looking at. So this is carbon sequestration at work. This is, and it's also food and home for an entirely diverse, entirely interesting set of fauna that we are not looking at on this trip, but many other people do, and it's pretty cool stuff.
Yep, I agree.
Thanks. Can you tell that it is whatever ungodly hour it is?
No, but that's a good point. We kind of go quickly across all of this, but there are many organisms that live in the sediment. There's a whole class of biologists that study those communities. It's called meofauna, what lives right
Yep.
in the surface of what we're seeing. Often pretty small, and some people, like us, aren't spending our time looking at those, but sometimes when we come out with really fully interdisciplinary groups, we have a group that would be studying the meofauna, and we would be attempting to take push cores in order to collect those samples, as well as trying to do more of the macrofauna, so the corals and sponges. But for the time being, we're just gonna keep traversing until we come to more hard ground.
Yeah, it's difficult to speed up too much. With
No, yeah,
with
you're
the
good,
ship,
yeah.
because
Understood.
And even though we are not multidisciplinary enough to have meofauna biologists on board, we do have a diversity of different types of expertise on board, and it takes everyone to work. So, for example, Alexis and Steve, I think I'll call our true coral experts. Steve is an expert coral taxonomist. Alexis is an expert deep sea biologist with a focus on corals as well. Tim Shank, sort of seamount deep sea biologist extraordinaire with a heavy focus on coral associates. Brian is starting to think about sponges more and more. I am an ecologist, and I think about processes in all kinds of ecosystems, not just the deep sea. So, believe it or not, even though I'm chief scientist on this expedition, I'm learning from everybody else here, which I think is part of the collaborative fun, because everyone brings different expertise to the table. Anna's a microbiologist, but really an immunologist working with microbial communities. Ana Merde is a beginning marine biology student. Constance is an artist who has expertise in shallow water corals and thinking about oh my god, what's it called when you take one thing and you switch it?
Transplant
Thank you.
experiments.
Oh my god, I'm really tired. Doing transplant experiments in shallow water reefs and looking at transcriptomic responses. So we really do have a diversity of expertise on board, and I'm very grateful that we have all of these different voices and expertise is on board, so that when somebody doesn't know something, the likelihood is that someone else does. And I think we're able to run a really interesting cruise and do some really novel science, partially because of that interdisciplinary nature, as well as sort of checking the traditional check boxes, which needs to be done, of basic exploration and characterization of the seafloor, taxonomically, as well as ecologically, starting to probe into some of those more process-based questions, and then experimentally doing a bunch of work up in the lab. And that was the longest run-on sentence ever. But I'm pretty psyched about what we got going on.
Yeah.
You too? Yeah. It's
And
good.
we have a pretty small science team, somewhat due to COVID, so we had to kind of have a very select group that was able to, everybody can do multiple things. We have 2 people on watch at a time, which is pretty short-handed compared to other operations. And so we all need to be able to contribute in not only doing the control room and ROV work, but also in the lab in multiple ways, and also, working to plan the dives and do kind of the background work as well.
Yeah. I'm just thinking about the interdisciplinary nature of what we're doing too, and I think one of the
Okay.
yeah, let's pick up another piece
Oh.
of trash. Yuck. And one of the other things is that I have a lot of experience working in this part of the world. I've been working here for 15 years, and not just in the deep sea. In fact, the deep sea is a relatively recent part of our repertoire. We've been working a lot on the shallow water oceanography, the shallow water reefs.
Can we look at it first?
Let's get a good snap zoom. And it's important. One of my goals is to sort of put all these various ecosystems together, to break them out of their silos and sort of get a more integrated view of how they work. Unfortunately, some of that integrated view is, I don't call it unfortunate, but it includes a look at history. There's a microplastic that came off of it. See, the little green blob to the left. What we're looking at here is some debris that's left over, probably from World War II, would be my guess.
Yeah, I can't. I know that there was activity here around then, but that's.
Yeah. So you can see to that left, that little green dot is it obviously came off of what we're looking at, which is most likely part of a shell, I think. But you can tell just by its size, that little green piece, is that it's now becoming part of the microplastics, which are defined as anything that's sort of less than 5 millimeters.
No, it's made out of it'll be a mixture of brass.
Brass.
Yeah.
Oh, it is brass. It's not. It's not plastic. Really? It looks.
don't think
The
so.
way it's broken, it looks plasticky.
Yeah, it'll flake off because it's coated.
Oh wow! Is
I mean,
plastic
if it's
coated?
from the.
No,
If it's
Is
from,
it brass coated?
It's just the way the
You
brass.
think of it from.
There you go. That's
World
how
War
little I know
II.
about it.
Yeah. It's gonna
Well,
be metal versus.
I wasn't even thinking about that.
Yeah.
Of course it is. So it's
Newer
not
ones-
plastic at all.
Newer stuff will have plastic.
Right.
What?
Newer
Yeah,
ones.
newer
Yeah.
That would be
stuff.
plastic,
It
but.
It looks plasticky, but let's pick up some trash and at the same time learn something about what it's made out
Yeah.
of.
It will be pretty cool.
If it's newer, then we'll know. But goodness, does it feel hard? Can you tell? Good. So it's metal. Okay.
Oh, okay. This is, I guess, gonna be
What sample number is that?
251.
Oh, we don't have an other either.
Oh, sorry. I guess we never expected.
It's trash. I'm gonna put it in as sediment. That's going into the rock box.
Yeah.
Also, I apologize. I think I forgot to list our last trash collection as a collection, so it's not going to be in the sample.
Okay.
Could you make a note of that in there so that we
Yeah.
have it? It didn't occur to me that it could be. It didn't process as a sample.
Yeah.
But
Yeah, yeah.
again, I'm so tired I can't even think about metal versus plastic in World War II. So don't trust me for anything right now.
She's camping. She does it a lot.
So it was a micro metal.
Yeah.
Micro metal.
Got some Xenos.
So that's the second one of these we've seen. Halfway down.
Almost done, but there's a problem.
Oh, we have rambling.
There's Diane. Hi, Russ's mom.
Russ,
Hi,
that's your
Mom.
mom?
How's it going?
Aw, cute.
Aw. I love it when family
Yeah.
friends and family watch. It's great.
It is fun. My other friend sent me a video of them watching the video.
Oh, I love that. You know, here we are in the middle of the Pacific Ocean,
I know.
right? Like we're basically right on the equator. There's no one else around us. We haven't seen another human for days and days and days and days
Except
and
for the
days.
39 of us.
39 of us on ship, and yeah, we can talk to our friends and family by having, and all of you by having you watch this live streamed. It's pretty amazing.
It is. What time is sunrise?
It's not a fine, but
Got
Yeah,
it.
it's probably just they got distributed, right? Like,
Yeah,
I mean, we're 2,000 meters.
falling down the slope.
Well, that's a loaded question.
See, look, Randy.
Oh.
Like, this looks like what we're. I mean, I don't know the sizes of those,
Right,
but like with that base, you know?
right, exactly. It didn't, and it,
You just have to go at this.
Alright.
Just the A is too much.
It's just after however many years in seawater.
exactly. It's amazing though how it looks. So it really does. It's amazing. It's so different.
Yeah, it's like all
Yeah, yeah.
crackly.
Exactly.
Oh, it's not.
Nope. That's not it. Thank you. That was
Mhm.
helpful.
Just having sorry about it earlier, so just can't anyway.
Okay.
Alexis, do you want to answer this question?
Yeah. There's a question of are we looking for something or just cruising around? We're always looking for something. We're on a dive track right now, working our way up the side of this island, and we're really looking for deep sea coral and sponge communities. which would require hard bottom or like rock surfaces, and so we're in an area right now that is relatively sedimented, even though the slope is really steep, so it's somewhat surprising to find that. So we're kind of cruising through some of that type of habitat, I guess, to look for areas where we would more likely find these larger communities of corals and sponges.
When we find these corals and sponges, we have a list of objectives, including characterizing them, and in some cases describing them for the first time. There's lots of new species and new morphologies and range extensions and things like that we've been finding, in addition to looking at the coral and sponge associated invertebrates, as well as looking at ecological processes of corallivery and predation, as well as looking at the microbiology, as well as looking at eDNA in the area, as well as geologically characterizing the seamounts, as well as looking at, we have a big agenda, immune responses of ancient immune responses in these basal metazoans like corals and sponges, and so on and on and on. There's a big agenda, but first we gotta find them. So we're hiking the sandy terrains of this very steep slope where we just, if you are just tuning in, prior to this we were on these incredibly bouldery rocks, so a lot of hard bottom, but just because something's hard bottom or steep, which is where we expect to find corals and sponges, doesn't mean they're always there. So we usually move through these habitats and then find what we're looking for, and then sometimes the habitat changes again and we just have to move through it and find what we're looking for. So it's
Then-
always this is the nature of exploration. We're on a track that no one's ever been on before.
What's our
You know,
shallowest waypoint?
Even though other people have looked at Baker Island before, we're looking at seafloor that's never been observed before. We're on a track that has never yet been explored.
1460 something.
Yeah.
Oh, okay.
1455.
That would explain why strength.
And we're at 2,200 meters now. We're working our way up to around 1,400 meters. So it's also when we have the chance to what we call make tracks, so move more quickly. That just gives us more time when we do find an area that we're more interested in spending more time. Today's dive is probably
I teared the depth.
what we're coming out at 1.
I'd like to be
5,
on top at 1.
so like 5 more hours.
I don't know what time it is. Yeah. I'm so tired.
Yeah.
Yeah.
Or, no, 6 more.
6 more hours.
6 more
I
hours.
teared depth there when we were on deck.
Thanks. Yeah.
Weird.
And this isn't, this is kind of what real exploration is like. If it's somewhere we haven't been before, we don't know what we're gonna find, and sometimes it is, it's patchy. You have patches of areas where you want to spend a lot of time, like some of the spaces we found yesterday, that it's hard to pull yourself away from, and then there's other times where you see an area that is devoid of corals and sponges, but it's all interesting and valid information for trying to understand the overall characterization of these depth ranges.
We're in a no-take protected area as part of the United States Pacific Remote Islands Marine National Monuments, and every little piece of seafloor that we image helps to contribute to the monument's overall understanding of what the U.S. is protecting here. And it was the same thing when we went to the Kiribati Phoenix Islands in 2017. It was the first time between Okeanos Explorer and Falkor that we were able to characterize. I have it. I don't know what it is, but I clicked the fish button.
That's okay. We know it's a fish.
It's a fish. And so, and the Phoenix Islands Protected Area is also a no-take protected area. So these two neighboring MPAs, marine protected areas, are really important because they cover a relatively large area in a really important geographical position where there's a ton of exploitative fishing around us, which, so that a lot of our tuna comes from this part of the world, and so protecting spaces that are large enough that can maybe help to bolster or improve tuna stocks is a really important replenishment mechanism to keep sustainable fishing in the oceans. But trying to understand, fish don't live alone without an ecosystem around them. They use everything. Everything's connected, and so trying to understand the deep sea flora, which is frankly the vast majority of the benthic habitat that's here, is part of understanding what makes healthy oceans, and so we're characterizing it to try to contribute to that.
Yeah, and one component to that is pretty interesting that Brian had talked about earlier in the cruise when we're not quite so busy working with the ROV, we do little seminars at sea, and so Brian talked about some of his work and thinking about the different types of communities that you find on different underwater features. So you have seamounts, which are completely submerged underwater, and then there are atolls and islands like what we're on now, where you have actual land at the top of the mountain, essentially, that breaks the surface. Emergent
features versus
yes,
submergent
there you
features.
Go. Versus guyots, which are seamounts that are flat at the top, so at some point they would have been eroded away when they were at the surface. And so the more we can see of these different types of features, the better idea we can get if there's differences in the community structure based on the type of feature that we're on.
It's a really interesting, that's one of the pieces of work that, it's one of the first things Brian did when he got to the lab, and I'm really proud that we were able to work on that together, because I think it's one of those things where a little bit of shallow water insight was able to sort of help think about what might be happening in the deep sea, because I think, you're thinking like, I'm 2,000 meters below the surface on the side of a seamount, who cares what's above me? But it turns out it might really matter.
Yeah.
When you think about it from a shallow water perspective, of course it matters, because there's tons of stuff that's really different in one part versus another. But putting these, I think we often put ourselves in. I'm a deep water biologist. I'm a shallow water biologist. I'm a oceanographer, or whatever. All of these different categories, and while we can sort of speak to each other's ecosystems a little, it's really hard to work across them, and so that is part of what we are trying to do. But some of the insights I think are going to be pretty interesting because of it. So Brian did a great job with that paper, and it's in the Frontiers of Marine Science if you're interested in checking it out. I think it's called The Unknown and the Unexplored: Insights from the Capstone Expedition Across the Central Pacific, or something like that.
Look up Capstone.
And unknown and unexplored. That paper was a ton of work. Did he ever tell you the backstory behind some of the things that we did? In order to find some of the patterns, we printed out something like 180 pages of draft graphs. That was him walking in. I was like, oh good, he can tell us. And he and I, we had this long hallway in our department, and we taped them up over all the walls so we could look at them all at once, and then just kind of like reassorted them. We sorted them by depth, and then we sorted them by feature, and then we sorted them by latitude, and then we just kept untaping them and putting them around until we could sort of see the whole story put together. It was hours and hours and hours of storyboarding on the side of the wall.
That is funny. I mean, that's how you have to do it sometimes. You need to be able to physically move things around in order to get it all pieced together in your brain.
I know, but you see the final product and you're like, oh, it's a figure.
Right.
But there was like a million figures, like small figures that we had to literally, I just remember it was never time during the day because there's always too many meetings. And so after I put my kids to bed, we call it the mommy hour. I would come back to work.
Oh my gosh, really?
Yeah. We would spend hours into the evening moving graphs around until we could kind of wrap our heads around that story. Isn't that funny?
Yeah.
So I don't know. I think I'm thinking of it now because it's late at night.
Right.
I'm thinking about the process behind it, but it was a really fun paper to work on. Partially because of that, we don't always have to get so tangible about it.
Yeah.
But the printouts were very useful, critical.
I still have to print things out when I edit them. I need it all there.
Yeah. Yeah, it's really fun. And Brian being Brian, I think we have it somewhere, came up with the idea to take a GoPro and time lapse us
Oh my
doing all
gosh,
this.
that's actually really funny.
So it's somewhere.
I want to see that.
I don't
You
know where it
guys,
is.
like,
You'll have to ask him for it.
Scurrying around.
We thought about it halfway. I wish we thought of it from the very beginning.
Yeah.
But he thought of it halfway through and said, let's do this. So we have this thing up, and the two of us are like staring at this part of the wall, stroking our chins, like not faked, right? Like just,
Right.
we didn't even realize we did it, but it was hours of that moving around.
It's funny.
But it's funny.
Go ahead, Rich.
Maybe I should have some coffee. I'm
I
starting
know.
to ramble.
I was thinking I might need a snack too.
Go get some if
Yeah,
you
so
want.
what
Or
Let me
we're
just
saying-
run down
Yeah.
and I'll come back after that.
Behind
We can
us
go.
and closer to us because the locals get blown like a big loop behind us. We're thinking that might be allow us to bring it in closer and have more of a direct route. So. Yep.
So I just go touch. Yeah. Doesn't help us, it's 70-degree.
What's that?
It's a 70-degree roll on.
Yeah.
We have a little sea urchin. Maybe not so little actually, but hanging out in the middle. Here?
So, we're just adjusting the ship's position so we get a better
No, yeah, I got it.
We're getting cold.
So we are readjusting our position as we check out all of this beautiful and important sand on our way to find some undoubtedly amazing coral and sponge communities. They must be incredible, but we haven't found them yet. This is totally unexplored territory. Nobody's ever been on this part of the island before, underwater at these depths. So we are the first ones here. You're here with us, and we're traversing along the seafloor, checking out.
Look at this, just for 2 minutes. Ship train.
What?
So I'm gonna sit down with the ship.
Yeah. That sounds good. So we're gonna sit down and take a look at this urchin. Well, let me get into his position where I can help us zoom. We can measure this thing. So those two dots are separated by about 10 centimeters, or they are 10 centimeters apart, and so you can kind of get a sense of this. And it looks like there's a little invertebrate to the left of it, so it might be a little scale worm. That's what it looks like to me. You can see this urchin accumulating lots of little bits of sediment around it. It'll sort of clear it off on its own if it doesn't want to be covered, or the current will just move it off.
There you go.
No, it's just we need to get them done in. There's too much out. Oh, get an average. See what there's
We're in the middle
Is it?
of the three. Yeah.
seeing some shrimp as we cross the seafloor here. Oh, Alexis is back with some coffee. I think I'm gonna go grab some as well.
Debs, you finally made it to a dive.
I know.
She's
Look
so
at
happy.
this. Imagine this. An RV dug. Oh, this is what they look like. Yeah. Lots of sand. I was like, what's the Dumbo? Yeah, I saw I was in London. Four two three and 422. I was like, wait, there was an octopus. Wait, there was a fish. There was one of those orange frogfish.
Saw a ray.
Yeah, we had a big ray yesterday.
We were running for a train. Stayed on ship. Mass petty train.
I was talking to my niece earlier. She's 3. Showed her the board. What is that?
There's some rock.
What are you doing? Why are you looking at that?
Maybe we'll get
My
some
sister
more
said she
rock.
showed her the pictures of the trail shark. And she was like, I love that.
Oh, we have an anemone. Let's take a look at it. Because it's something.
See, I'm full now.
What's that? That happened to me yesterday. No, you're on lock right now.
Alexis.
Yes.
So you're unlocked at the top of the lock.
Ooh, here's a sponge. We can take a look at this glass sponge. Starting to see some more rock exposure, so hopefully as we go up we'll keep getting more exposed rock, which hopefully could mean more corals and sponges.
Nice
We've been
hexactinellid.
we've been working our way up this this very steep section of the island that we were hoping would be more exposed, so having less sediment drape on it, but sometimes this is what you get, and that's okay too. Oh, there's a little coral right there. We'll just quickly,
Anthomastus?
yeah, I think it's an Anthomastus. It's a little mushroom coral.
All right, I know I need this coffee because I'm like, where's Anthomastus? And it's literally the first button on the thing.
Take a quick zoom in. serotonin hit of seeing something other than sand. Nice, that's good. Alright, we can keep going.
There you go.
There's a sea star over there. Loads of stuff.
Well, I don't know if I'd say loads, but there's stuff.
It's a 200% increase in biodiversity.
Yeah.
What did you put in that coffee?
She hit the espresso button several times.
That's true.
You got another sea star up top?
Yep,
Another
there
sea
it
star.
is. Plethora of life. You know, never mind.
Okay.
See, we better see.
A rock.
With a dead sponge on it.
With a dead sponge. Hey, there's some sea stars down at, or ophoroids at the bottom though. Those are big. I'll just come in a little bit.
Are they considered non-associated if they're on a dead sponge?
I know, that's a good question. That also makes you wonder, like, whatever attracts them, they're still attracted to it even though it's dead.
Structure?
Well, but there has to be something that attracts them
Yeah, I know.
there initially, right? Alright, we can keep going. Oh, something over here.
Hard ground.
Rocks on rocks.
Huh?
Said rocks on rocks. There's, I think it's a dead stalk or something.
With the crinoid on it.
Yeah.
I love those.
Yeah, it's just a old base of the crinoid.
I can't even remember who I did what with, but I think it was it with you that we did the outreach event where we were talking about what our favorite deep sea
Yeah,
organisms were?
yeah. That was yesterday morning at this time. Fish.
I can't remember what you said.
I said octopus.
Oh yeah. I said crinoids.
You did. I think there's something over here on the left, Russ. Okay.
I wonder if that is an approaching coral wart.
Maybe. It looks like, this looks like that paragorgid with the zoanthids on
Yeah.
it.
This is
That same kind of balance that we've been seeing. I hadn't thought about that until this cruise, that this combination of coral plus the zoanthids. Normally you see zoanthids kind of take over a whole colony, but we've seen this paragorgia and zoanthid combination a lot, where you still have a lot of the paragorgia polyps that can still open and feed themselves. So it's almost like there's a balance between them versus the zoanthid being more parasitic, which is what we kind of consider them to be.
Is it an astroschema star?
Yeah.
I think. Yeah.
Yeah, it is.
And it looks like some kind of evoplasoma too, nearby.
Yeah, definitely. Not a cup of tea. Anyway, I've got-
So the pink
got a
coral
walk
underneath is a bubblegum coral, or a paragorgia, and then you have the yellow zoanthids, which are growing over the top of that.
Nah, it's little yellow.
and a big astroschema brittle star in the center. So this is a pretty standard combination of the three of them. And then you've got the sea star over here.
Yeah, let's image that.
So try and take an average between
I'm
the three.
tempted to pick it up just for stable isotopes, but I don't think we have enough from this spot to be able to really compare it. But then again,
I mean, we don't know what we're gonna get,
find.
right?
Yeah, maybe we should grab it.
Okay, that's it.
CTD.
We can take a good image of it.
Let's take
though.
some really good imagery of this.
Yeah, it makes
We can identify it. And look at all those little sponges to the left. They're so tiny.
Oh yeah.
I have to. What if this is spongivery?
Oh maybe, actually.
Hang on a second. Hold on. Because look to the left. Can you look right? See where this thing is? There's
You want me to
sponges to the
come
right
down?
and sponges to the left,
Yeah.
and where this star looks like it's climbing up, there's a gap.
You're right.
What?
I said you're right. I don't, but I would think that we would still see some bases. I don't know.
No, they're pretty good about wiping it clean. Hang on. I bet you this is spongivery. What kind of sponges are these?
Don't know. Little
They
spiky
are.
ones.
I mean, I think they're hexactinellids, but I'm not really sure.
Yeah, I'm not sure. I would just write long, thin, or short, thin, spiky sponges. Somebody asked what's the water temperature, and it is currently 2.1 degrees Celsius down here at 2,100 meters.
Can we get a scan of the whole spongy area and then see here to the right as well? I just want to kind of get a sense of how big this field of little sponges is.
Do you want lasers on it then?
Yes. I'm pretty sure this is spongivery.
Okay.
I mean, I can't tell for sure without collecting it, but this is looking likely, given that these are highly predatory sea stars that we know eat corals and sponges. And thank you, that was really helpful. And I thought originally it was advancing on the coral, but there was no evidence of corallivery whatsoever. But this is like a field of sponges with a pretty a clear strip with no
Oh, we
sponges
have a little
whatsoever.
urchin up there too. We'll zoom on real
Yeah.
quick. Oh,
And
another
isn't there
little
coral? Jinx. All right, that was great. Thanks, Alexis.
You're welcome.
Here's my other phone. I'm leaving my crap all over the place.
It's like the other day, Tim lost his phone, and we're like, well, it has to be on the ship, so.
It's true. Somewhere with our PCR films.
Yeah, yeah.
I'm so.
Oh, look at that astroschema. Is
Oh, it's off. That we should probably think about collecting. Let's
see.
take a look. There's been a lot of astroschemas.
Yeah. It's gonna be a little bit hard to. It's probably a different kind since it's not up on a coral, but it looks like it's missing a couple of its arms. Do you see?
I do. It looks weird.
Or parts of them at least.
I don't know though. Usually when they're that long arms, they're
They're on a coral.
on a coral. We can maybe take a look at the one that's actually on the coral and see if we think they're the same. The color is different, but it's not always.
Yeah, it's hard for that to be.
What kind of coral is that actually?
I think it might be a young precious coral. But it's hard to tell when they're this small. What do you think, Randy? Should we go for that
think we
lone brittle star?
I do.
Okay. Want to slurp it?
I think we should.
Okay. Alright, guys, can we slurp that brittle star?
Precious corals are correct. No.
Corallid.
They are corallids?
Yeah.
You thought
Corallid.
with that heart? Huh. Okay. It's very low. Very. Okay.
Oh, you know what? We should have taken a Niskin at that first coral site. And we forgot
Didn't even
to.
think about it.
I know. I think Steve said something, but I think we both were like, what?
We'll do it again. I mean, again.
I know what you mean.
We'll do it at our next spot. I was just saying to Adam, who's now here, sleep deprivation. I'm like, usually I'm pretty good with it, but this cruise is a whole next level for me. I'm coming off of a year and a half of pretty extreme sleep deprivation. And now it's, I'm saying things like, we'll do it again, like go back instead of go forward. But speaking of which, we've had a watch change in the ROV pilots, and we have Adam and Ben flying right now. And we still have Debs as our marine tech.
We still got Debs over there in the corner.
Debs hasn't had a watch change yet.
That doesn't have anybody to watch it.
We're so happy for the consistency of Debs. And so lucky. And then you still have Alexis
Hello?
and me. Hello, Randy. And we're setting up to sample this brittle star. Did you have a tough COVID year?
Huh?
Did you have a tough COVID year?
It was interesting. I was finishing my PhD, so that wasn't the most fun to do, and we were all stressed out about if we were going to be living or not.
But I have this very fond memory of watching your PhD defense outside. It was like a nice day.
Oh,
and
that's
I was
nice.
outside watching you, and you just did this beautiful job.
Thank
so that was fun.
you.
So yes, I'm sure the run up to that was hellish, but the
Yeah.
final product was beautiful.
Thank you. I had really good neighbors too at the time. I was in Philadelphia then, so that really helped. We had we had a big backyard or a big open lot behind our houses in the city, and so we would go out there and the dogs would play, and I think that kind of saved me because I was otherwise would be seeing no people.
See if you can get another quick zoom
Yeah.
before it, because I didn't take a
Can I
zoom
get
as a sample.
zoom real quick, Adam?
Yeah, go for it.
Thanks.
It's amazing how good people in good spaces can really replenish.
Oh yeah. It's a
Here we
little-
go. There's two of them actually. See?
Well, that's a different one underneath.
Yep. Okay.
Let's go.
Good.
In. I wanna. Wait.
Even better. Even better. I'm just taking 25 sample and CPs.
Great. Okay. Thanks.
We'll
This
chalk
is
that up.
going to be sample 252.
Um, 252.
Correct.
Astro schema on the rocks.
Yeah, I'm not sure if it's astroschema. Might be a different
I'm not sure either.
genus of. I just said ophoroid.
It's very little.
Just gonna try and get away.
tug of war. Nice job. That's going into which one? Let's see. 2.
I'm gonna kill it and start it again.
There we go.
Nice.
Is that jar 2?
Should jar 1.
Oh, sorry, jar 1.
And
Alright, that's all. Yeah, I like that for something fast. Is that always what we're doing? Just that's awesome.
Okay, we can carry on.
nice job.
Shall we proceed?
Yes, please.
Okay. Shrimp.
Bridge ROV. That's an update video for that. Yeah. Thanks, Tony.
When everything else moves so slowly, it's always amazing that they have so much energy to move so quickly.
shrimp?
Mm-hmm.
Yeah, I mean, they probably migrate, right? They're probably not only.
Sure,
Deep
to
sea,
find,
yeah.
find food sources, but it still is amazing that they're just so
Scooting along.
scooting along, never idle.
There's some sponge.
Something. Looks like there's a star on top of it too, but I can't tell what kind yet.
Yeah.
Let's take a look.
Just gonna quickly zoom on this bit of sponge. Looks like mostly dead, but a little bit live. Looks like a probably a spiny arm, so a few canted. Lots of little brittle stars in the dead sponge too.
Yeah, I think the little ones look like they're smooth armed, but I
Oh.
can't tell.
It's okay. We don't need to spend too much time.
No, I'm just
We can keep
marking
on,
them all.
keep carrying on. We'll just stay with the rocks, you know?
There's a big anthomastus.
Yeah, and a crinoid.
Speaking of your PhD, Alexis, you should tell people what it was about.
Oh my gosh. Well,
It was pretty awesome. It was a deep sea biology story.
It was. So I did my PhD at Temple University. Most of my actual dissertation research was stationed in the Gulf of Mexico. There's large colonies of Lophilia. We've got a
Got it.
glass sponge. Large mounds of Lophilia pertusa, which is a deep sea scleractinian coral. And when the Deepwater Horizon oil spill happened in 2010, it was relatively close to some of the large mounds that we knew already existed there. And when they went down to do surveys, some of the first cruises after the oil spill found signs of impact at some of the octocoral sites, so Paramaricia, which is a type of plexaurid. They found oil flocculent material, so like brown, mucky material on the corals. But when we went to look at Lophilia, there didn't seem to be much of an impact. We didn't see any sloughing or flock that had settled there. Looks like we have a paragorgid. Just gonna quickly.
Yep.
Yeah.
With an extra schema.
Yes.
I think so.
Yeah, I just am not sure about
Actually, no. Well, yes.
Sorry, I came in really fast. Okay.
Yeah, I think so. And looks like there's some zoanthids in the bottom of it. You see that?
Yeah. It's just the way the polyps are attracted. I'm not sure that it's Paragorgidae.
Huh.
Okay. Yes. It's very pinkish.
Why do you think it's not paragorgid?
Just the way the polyps are retracted. It doesn't have they're not retracted into the choanenchyme tissue. They're sitting on the outside. So let's just take a closer look.
There you go. Oof. I see what you mean.
See what I mean? And they're kind of barrel shaped, sitting on top
I totally
of the
see
tissue.
what you mean, but I don't know what else it could be.
It could be a corallid.
It looks so flexible. Maybe not. I don't know.
I mean, we can just note that it's either or.
Yep.
Whoever is doing video analysis later can
We'll look it up.
can't seem to get it to focus. I think it's a acorn worm, maybe.
Is that an acorn one?
I think so. It's just very transparent. Normally, or sometimes they're like a purple. The top of it looks very strange. But it does look like an acorn worm. Just has like those, I don't know what you call those, the flappy parts.
Yeah. Hang on, we don't have a,
Acorn worm button.
no, we have a misc worm button. So, hang on, can you go back into that for a
Oh,
minute?
I'm sorry, yeah.
It's okay. I just was finally getting to look at it. God, it looks almost like an orchid.
Yeah, yeah. It's very dark in that one region of
Yeah, I wonder if
gut.
that's... it could be reproductive organs, or it could be gut right there.
Huh?
So it could be gut, or it could be part of the... Sometimes they have reproductive organs right in the
mhm.
same sort of region. I don't know. Very cool. You know, they're acorn worms, but I always think of them as arrow worms because they always look like they're pointing in a direction.
Yeah.
Like, this way, this way.
They leave those trails
Yeah.
as they crawl along. Okay,
You
guys,
can
I
see
think we're
it's-
ready to
it's
carry
track
on.
too, see?
Yeah.
Yeah. Where are we? 2,088 meters.
2,088 meters.
Almost to waypoint. Halfway past waypoint 2. Sorry, that made no sense. Just past waypoint 2.
I know I'm drinking all this coffee now, and then it's gonna be 8 a.m. when I'm supposed to go back to
I know.
sleep.
That's,
I
I'm only
won't be
having
able to.
one cup, but I'm hoping I'll be able to go back to bed.
I'm sure we will at some point.
Stay awake for morning meeting, make a plan, and then pass out for a while.
But yeah, from my PhD, we
Oh, yeah.
we found these. I know. Let me get back to my story. We found these. We wanted to run experiments to understand if Lophilia was impacted by oil or the dispersants that were released during the Deepwater Horizon. And so I ran the first set of experiments was just exposing the corals. We went out to the Gulf of Mexico, collected live coral, and then went back to shore and shipped it to Philadelphia, where we had tanks set up for cold water corals and a cold room where we could run experiments. And so I ran the first set of experiments exposing them to different concentrations of oil and dispersant. And what we found was at a higher concentration, which was based on what we saw in the sediment around the coral mound, that Lophilia wasn't really stressed out or impacted by exposure to oil alone, but when you added the dispersant, and especially dispersant alone, they were very stressed out by that in a relatively short time series. So we took measurements every 12 hours for 96 hours, and then I also was then interested in if an oil spill happened in the future. So if we change the pH and the temperature of the water for what it might be around 2020, or what's predicted for 2100, will the corals respond differently if they've already experienced heat stress or more acidified water, so a decrease in the seawater pH. And when we ran those experiments, we found that the pH didn't really have an impact on their overall stress level or health, but there's a crinoid. But when the corals were exposed to the oil and dispersant under increased temperatures, that they had a harder time recovering from that exposure. So they still experienced increased stress during the dispersant exposure, but when we tried to see if they would recover afterwards, we don't need to stop for that. Yeah, thanks. When we looked at their recovery, the corals that had been exposed to warmer waters had a harder time recovering from that stress that was induced by the dispersant. And we looked at that on a phenotypic level, so how is their behavior changing, and also on a transcriptomic level, so how is their gene expression changing? What genes are changing the amount that they express during the different exposures? And so we saw those kind of general results of the dispersant being more harmful across both the phenotypic observations as well as in the transcriptomic data. So it was a really fun study. It took a lot of experiments, a lot of time in a cold room, but we found some pretty interesting results, which is always nice.
You know, I think, so Alexis has a beautiful paper in Scientific Reports, I think is where you published it on this. And it's really important work for a couple of reasons. One, obviously, it's super applied. Two, it's a lab experiment, and those are rare with deep sea corals. And then three, it's sort of getting at these subtle sublethal effects, which are not super obvious to look at, but they're very important, because I think if you're just looking for the 2-second punchline, you know, coral dies when exposed to oil and dispersant. You know, it's not that simple, right? It's corals are stressed, and they have all of these subtle impacts that'll last, you know, a really long time, and that's part of this too. These corals are really long-lived, and so whether or not these exposures impact them for just a little while or impact them over their lifetime, which could be hundreds of years, is a really important question that Alexis obviously can't. That's outside the timeline of what you can do in a PhD. But you know, it's these sublethal stressors are often ignored because they're subtle. They're more subtle, and they're not as physically obvious, and they you know, the punchline, they're harder to capture in a 2-second punchline. But I think there's a lot of sublethal sort of slow stress that happens to organisms when they're exposed to anthropogenic influences that is often unaccounted for because it doesn't show up as sheer mortality.
Looks like this is a sponge, if we can take a look at that.
Good idea.
Yeah, and that's a good point to what Randy was saying about sublethal effects and really kind of small, what seems like small, minute changes. That's why we did the phenotypic part of the observations, was to see just how they were behaving. Were they happy or were they sad, is kind of how I sometimes talk about them. Because what we observed was that it takes really looking at them very up close to see those changes. So if you're going to a place that's been impacted by an oil spill or something, you might not observe those types of changes unless you are, you know, at max zoom looking at a whole big mound, which is not really conducive to doing ROV work. So that's why we do the genetic work. It is right next to the sponge.
I don't know. Is that a pycnogonid?
Let's see.
Yep. Sure is. Well, I think so.
I think it's some type of crab.
The legs are wrong for pycnogonid.
Yeah.
6. What kind of crab is
Or
that?
is it a picnic? I don't know.
Brian, you know what that is? Brian Kennedy's just walked into the room, and he's got a ton of deep sea experience from all over the Pacific, and got a ton of really great general knowledge. So maybe he knows what this is.
The only thing I think of is isopod.
Oh.
Isopod?
Yeah,
It could,
long
yeah,
isopod.
could be
Yeah,
a type
cool.
of isopod.
So my first guess was definitely sea spider, but I think isopod is probably more likely.
Another long-armed isopod does not have a button, just so you know.
Well,
I'm not complaining. I'm just trying to be funny.
I know.
The short-armed isopod. No, it's an amphipod.
No, I don't. I think that's a.
No, it's an amphipod.
No, Oh, that's an amphipod to the left, but I think there's a spot lobster in the back.
Yeah, there is a little hanging onto the rock. Let's take a look at this again.
Hold on,
'cause it
I have
looks
to
like-
mark this again. Because now there's all kinds of things. Amphipod doesn't have a little list either. We should add an amphipod button. Could we make a note of that, Alexis?
Yeah, I mean, I keep saying yes, but I need a list of things to add.
Here, I'll put it in Slack.
Look at its little swimmerettes.
Oh yeah, look at that.
It's fun.
That's different. I've never seen one quite like that before.
Nope, it's a very cool. We got some really good imagery of that, so maybe
Yeah.
somebody can help us identify that. Those of you in the chat are experts at helping us crowdsource our
All right, I
IDs,
think
and
we're
so
good,
if you
guys.
want to stick that on Twitter and see what we get, that would be really helpful. Because none of us really know, which is always part of the fun of deep sea exploration.
What were the other buttons we need to add? Do you
I don't
remember?
know. We'll as I remember them, I'm gonna start sticking them in
yeah,
Slack for you
that's
now.
good. Yeah.
There was Kid the Gorgia Day.
Yes, that's one.
Brian, I think we saw some spongivery earlier.
Did you see there were, the unknown explorer had said something in the chat about Chris Ma responding to her on Twitter about that?
No.
Said what? Deb, can you scroll up in the chat just a little bit? Chris Ma said that the sea star eating a sponge earlier looks like Mediaster,
And yes, I'd say so.
as if
Oh,
it was
good.
eating the sponge.
Great.
Cool.
Spongivery. Thanks, Chris Ma.
And
Chris,
the
I owe
unknown
you
explorer.
I owe you a message. I'm sorry. It's been a busy 24 hours. A really busy 24 hours.
Yeah, he's probably just on Twitter. not
So
actually.
Chris Ma has been really helpful. Tim and I have been pinging him constantly, trying to learn something about these sea stars, and he's been incredibly responsive and collaborative and just wonderful. And I owe him a message. So Chris, if you're listening, I got you. It's coming. When it's not 6 in the morning, I need to be able to put something coherent together.
Huh? Oh, thank you.
Thanks, Lisa. Lisa's gonna toss that crab up on, or what we think is a crab up on Twitter for
Or
us.
isopod something.
Right, long-armed isopods.
Not a crab.
I don't
Sorry,
think it's
not
a
a
crab.
crab.
Yeah.
That's from crabs.
All right. We have.
We have another
urchin.
paragorgid or something.
Let's see what it is.
Yeah, we'll just take we don't need to sit down. If I can just zoom a little bit. And you can come in. See, that looks like paragorgia to me.
I agree.
And the other one didn't have the same polyps, right?
Wait, yeah, because these have the sort of the knobby-ended polyps. So
Right.
it doesn't nearly look as blocky.
The barrels.
Yeah,
aren't there.
exactly.
Okay, that works. With an
I'm
astro
pretty sure this
schema.
is, yeah. Marked.
Great, thank you.
Long-armed isopod.
Yeah.
So cool.
It's also interesting because it was almost translucent, so
Mm-hmm.
it's possible that we're passing over more of them. We just, you
We're
don't
not
see them
zoomed in
unless
enough
you
to see
zoom,
them. Yeah.
which is fine.
Yeah, what if this is wall-to-wall carpeting of long-armed isopods and we can't? I'm just kidding.
I have been somewhere where it's been like a blanket of very tiny, clear holothorians, like on sediment.
There it is. Okay, so it's a.
Munopsidae isopod.
All right, let me
Thanks,
append
Jason.
that. Thank you. I like this comment feature where you can go back afterwards
Yeah,
and add.
it's
It's
helpful.
really, really helpful.
And it's helpful because when we print out that sheet at the end,
Yeah.
it's in there. So if you, because yesterday, like we had a quiver mix up,
Oh, that's great.
but we could see
Yeah.
it.
What is that?
I think it's a hermit crab in there.
Yep. We should get a hermit crab button too.
Okay,
I'll add it to your list.
thanks.
We just have regular crab, but really, we should have a hermit crab button. What time is it?
6:30.
Do you want your spot up here, Brian?
I'm happy to go up there if you want to take a break, but I'm also happy to sit here.
I'm having fun with Alexis, if you don't mind.
Huh?
I'm having fun with you.
Oh yeah.
I have a morning meeting in 45 minutes. I need to stay awake.
Yeah, you
If
need
I-
to stay
If I go to
up.
bed now, I'm
No,
out for a
yeah.
while.
It's not a good plan.
No. But I will go to bed right after that for a while.
Yeah. So if you're joining us recently, We are at about 2,550. Wow.
It's
Two-
not
Yeah, let's take a look at this real quick.
2,056 meters.
Yes.
Got you.
On Baker Island in the Central Pacific, which is part of a U.S. territory and a protected area. Just gonna come in a little bit, Ben.
So that,
That looks like hemicorallium to me.
yes, with polyps out.
Correct.
And
And an
Astrea schema.
schema.
And a little oaf that's falling down the side. Swim away.
Okay, that works. Thank you. I know it took me a while, but I figured
that's
it
good.
out. We have another coral.
I thought it was yet another cool little lingo that I wasn't cool enough to figure. Which is true, but very localized. Alright, we got a coral.
Yeah.
This is very hard to see when it's got this planar view, but we're getting there. Thanks to our awesome pilots. We've got Ben and Adam flying.
Just come in on this.
So that also looks like a corallid to me. No?
Yeah, it does to me too. We're just gonna. There's some.
Is that a
gall.
snail?
Now we turn inside.
Would you mind? That would be great. Great, thank you.
Okay. That looks like
Yeah, definitely a corallid.
and
Oops, I think we're moving a little bit. It's okay.
Pinocchio, I mean an Astrea schema. And then what is that?
I know, there's some type of gall down here that we want to look at, right?
Interesting.
Um,
How much do you know about coral galls?
not that much.
Are they induced the same way that like plant galls are by insects? Like, is there some? Are they induced
There
by
might,
some kind
some of them might be, yeah. So I've seen, in other corals, like, can I have manual zoom? Or do I have manual zoom?
I don't know.
Right before I left, though,
Oh, I do? I'm sorry.
wasp-induced galls on oak trees were starting, and they are tremendously cool.
But it is that sometimes, yeah,
Yeah.
like something lands and then the coral grows over it.
Yep.
Um,
One looks like a giant tooth.
and this is the coral that also, there's a polychaete worm that will burrow into the skeleton. What we collected yesterday, we were able to see there was a whole lot of polychaetes inside the skeleton when we brought it to the surface. But it doesn't seem to harm the coral in any way. It just makes their...
Do you see the polychaetes now?
No, I
don't see any.
Not on this one. But they're also really, they're like inside.
Oh, yeah.
So it's hard to see until you have it at the surface. Okay, great. Great. We can
Here's a
carry
big one
on.
too. Pretty big coral.
It's a nice one, yeah. Tibbs up.
Huh? I have to stay over 12 straight hours.
Yeah, I just didn't wanna.
What?
Oops, sorry, that's my fault.
Hey Brian, do you have that GoPro video of us putting all those pieces of paper together on the wall? I was telling Alexis about it. You should show Alexis. It was kind of funny.
So he was telling me about your process.
Her process.
You went along with it.
I don't know why.
You enjoyed it. You took a GoPro video of it.
At your request.
No, it was your idea. It was totally your idea. What's that? I think that.
I don't know. Sorry.
Dead sheep hen stock. As I yawn my way through that. I thought that was a dead sheep hen stock.
Oh, it might have been. Looks like that's probably a sea cucumber. Or maybe a sponge.
Maybe. Let's see what it is, and then I will. Actually, I don't know.
Can I just quickly look at this, Ben? Shouldn't take but a minute. I think it's a cucumber.
I think so.
Yeah, it is.
Yep.
It's
Yes,
very
I agree.
beige colored.
I agree. I don't think we've seen that one.
It's like undulating.
It's dancing. Sea cucumber style.
Okay, thanks. I can keep going.
Swaying in the current, maybe.
Yeah.
Yeah, it's very unusual. I haven't seen one like that. That's cool.
I don't see that on this hard drive. I'll forward it at some point.
Huh? Oh,
I don't see that one.
that's too bad. It's funny.
Looks like there's another one of them. Beige sea cucumber.
Yep. Good eyes. I wonder if all those little mounds are it's
Oh. Excrement.
excretory piles.
Yeah. I think of them as having trails, but I guess maybe.
I mean, it looks
Yeah,
pretty,
yeah.
yeah. Yeah, I'm just looking at the picture of it.
Oh yeah.
I'm pretty sure that's what it is. With no knowledge about this whatsoever, it just looks like a
I mean,
most
it's
likely
very
explanation.
different
Yeah.
sediment pattern than
Crinoid?
whatever else.
Exactly.
Yes, crinoid.
Here we go. Little crinoid. Or not so little crinoid. It is amazing, again. The corals that are here are just great, but there's not very many of them.
Yeah. It's one of the main questions that we're always thinking about is why are corals in one place and not in another?
It's one of Brian's main interests, so maybe Brian will crack the mystery, crack the code for us. No pressure.
We hope everybody is having a good day wherever you are. For us, it's about 6:30 in the morning here, so we've been. This dive has been underway since I think they
12:30.
around 12:30. So 6 hours. We'll take a look at this coral. Looks a little bit different than what we've been seeing.
You can turn sideways.
Yeah, please.
And you never know what you're gonna get. We've been on Baker. We've had some pretty spectacular dives. This one, you
This
know,
is
all
fun.
signs pointed to spectacular, but it's not unspectacular. It just is. It just is not exactly what we had expected. But you know, as we get to the top, you can always. That's the thing about exploration. You can just turn a corner and get surprised at any moment
Mm-hmm.
with the giant primrose forest.
Yeah.
Let's see.
So this looks like a white something.
There's zoanthids in it again.
There are zoanthids in it.
Is that another? Corallium?
Let's see.
No, it looks maybe a little too soft. I don't know.
It's hard when the polyps aren't retracted to tell.
Well, here's there is a retracted polyp down there, if that helps you, but not very many.
Oh yeah.
See it?
I'm trying to see if it's moving or is it just the tentacles that are moving. Is the whole colony swaying?
No.
Okay. I would go Corallidae just off of this, but
That's what I think too.
it is hard to. Yeah, there's little barrel polyps. You're
Yeah.
right.
Mm-hmm. Okay.
Great. Big astroschema, like big astroschema.
It is huge.
Some of the ones that have come up from the Plexarids in the Gulf of Mexico are like scary big. Yeah, like
Wow.
arm span of like my arm span.
Is everything a little bigger in the Gulf of Mexico? 'Cause it's more productive, no?
Yeah, it's I guess more abundance. There's a higher abundance of things. They're not always that big, but there's a lot of different Plexarids. Black corals are there too.
Fair enough. Oh, if only we could ski underwater. Look at that ski slope.
You'd be going a little too fast for my comfort.
Skiing?
Yeah.
I love
It's
it.
very steep.
Are we going too fast?
No,
No,
I'm
no.
sorry. Ben, you're doing great. We were talking about
I'm talking about
if we
skiing.
could ski underwater.
Ski on the marine snow. It's a funny thing to think about in the equatorial tropical Pacific. To go
I mean, there's
but look at the
water
temperatures. It's
ski.
appress It's 2.1 degrees C. We're on there's snow.
There's-
We're on a slope. It's finally groomed. Come on.
It is finally groomed. It has not been disturbed in forever.
I don't really ski this late in the season because the rocks are showing.
Ah, nice, Adam.
Although I don't know, you might be able to kind of go around the patches and sands.
Could be like, what do you do when you moguls?
Moguls. I guess this might be
We'll
the
take a look
equivalent
at
of
this sponge as we come up to
deep sea glades, Adam.
Like sand dunes.
Let's check that out. not look like our normal shrimp friend.
Nice hexagonal, but yeah,
There's
there's
a crinoid on top.
something inside. I don't know if we're gonna be able to really see what it is though.
You want to look inside?
Yeah.
Bring it closer.
Or let's just take a look at the outside real quick and then we can do that. Yeah, let's try and peer inside the sponge. Ugh, my back is sore from the hunched over tube riding
Yeah.
last night.
Sorry I didn't stay in help longer.
Oh, it was fine. We didn't actually go that much longer
Oh,
after you
good.
left. Yeah.
I'm happy to do that every time if I have extra
I
time.
know.
It's no big deal.
But
I'm happy to.
well, and
when
it's
I
also
can.
we've kind of gotten to a flow of like how both of us can be processing versus trying to do everything together.
Right, that helps too. I mean, I won't always be able to, but I'm happy to when I offer. I mean it.
Yeah, I know. Yeah.
Okay.
You have this camera too.
Oh yeah. Okay. Can we zoom on the sit cam? I think it's just sediment, honestly.
It's sort of tubular sediment.
It's okay.
Where are you looking inside?
I'm trying to look inside the sponge.
Right in the middle.
There you go.
There. Actually, just iris up there.
Yeah, I think it's probably just like
Yeah, I
sediment
agree.
buildup.
I mean, they call these things V sponges, right? For a reason. And in this case,
they
we have
catch.
it catches. It's got. It's V's
There's
with
a
a sediment
crinoid right at the
inside and a
top.
crinoid in the top.
And
And a
crinoid on the side.
crinoid in the top.
I think that's good. Thanks, guys.
Okay. Let's go exploring.
Exploring. Brian's like, I can't handle them at 6:30 in the morning.
He's like, I just had this peaceful,
Right.
solitary water filtering experience, and now there's a lot of, I don't know what this is, but somebody likes it. Thank you, Jason. Jason likes our commentary. This is what this is called. This is called professional camaraderie, keeping each other awake on the seafloor
Yes.
at some crazy hours.
I don't think that one had shrimp in it. It didn't have the top to it like we have with the. I think it's euplectellid are the ones that have that
Yeah.
matrix across the top, so the shrimp stay inside. It looked like it just had some sediment buildup.
Yeah.
I think this is a predatory tunicate. Right here.
Yeah, I agree.
Which we probably don't have a tunicate button, do we?
We do.
Oh. Try yourself. No, it's okay. I did something correctly.
You did a lot of things correctly. I'm just adding to. You built the ice cream. I'm putting the cherry on top. That's all. We just need
Can we
a
put whipped cream on top instead
a
of
little
it?
acanthogorgid on top, a little amphipod, whatever other buttons we need. Oh, somebody else likes us. See, it's the Alexis Randy deep sea early morning comedy show.
Early morning, not many corals to look at.
How many
Comedy
corals
show.
look at comedy show? We've had a couple.
Yeah. Ooh, there looks like there's a little sea star over here. Can you take a look?
And something to the left of it.
Yeah, I'll take. This thing always is a
I
little
know.
beat
There's
up.
another one if you want it. I don't know where it went. Here it is. I think this one's a little bit finer. As a connoisseur of pointing sticks, I like this one better.
Oh, I think it might be a chitin.
I think it is a chitin. And a urchin. Look at this, the trifecta. Chitin, sea star, urchin. Let's do it.
Okay.
We don't have a chitin button, however.
Okay.
You were so close. I don't think we need a chitin button.
It's
Can
a
fill
nice
that in.
little sea star.
It's a nice little chitin. That is very cute.
Oh, it's like
It's got, you see that it's got like kind of along the edges, the around the ridges.
bristles.
Yeah. It's fuzzy.
Right. Again, and the fuzziness is what's moving. Is that?
Yeah, I agree. And I think it's, I first I thought it was just along the arms, like along the central
No, it
ridges,
looks
but I
like
think it's the
it's-
whole entire thing
yeah.
sort of is covered in fuzz.
Well, echinoderm, right? Is that spiny skin?
Yep.
And then we have this chitin over
It's
here.
very cool. It's opalescent. How's that for a word at 6:30 in the morning?
That is a great word.
Thank you.
These are really suctioned on to the rocks.
Taking those before.
Yeah.
This is, it's really, it's a really ancient body plan. It's very, very, very successful.
Yeah.
This body plan has been around for a long time. And there's
Where was our other?
urchin on the top. Just
Just to round it out. Oh, 2 urchins. Remember we saw those groups of urchins? Do you remember that?
I do. We
I
saw
don't
the
remember
bunch
what
of them
dime
together.
that was.
Look, actually, there's 3 of them.
Yeah.
But they were all
But
right
they
up
were,
next
like,
to each
right-
other. They were
yeah.
like a row of pom
It
poms.
must have been like a mating thing.
I don't know. I don't even know how they literally organized themselves that way without getting in each other's way. Right? They had to really move their spines around to coordinate
Yeah.
that.
Yeah.
They're more flexible than they look, but there were 4 or 5 of them in a row, right up next to each other.
Okay. Can we carry on?
Carry on. So there's a question: have we ever come across something that you just absolutely did not expect to see? To that, I think I'd probably say all the time. For example, just the other day, we saw these rows of urchins right up next to each
Yeah.
other, and like a little string of them. Like they were beaded, like really, the round bodies adjacent and touching, and I don't think it wasn't like stop the presses, got to get on the cover of the New York Times, but you know, it was unexpected, and I still don't really understand what was happening, and you know, I think there's little surprises around every corner, and I think dive by dive, right, those little mysteries start to, you start to notice some things again and again, or some things you only see once, and they sort of stay with you in the back of your head, and you think about them and process them, and sometimes figure out the mysteries, and sometimes
I found
the
a
mysteries stay
plane.
mysteries for a while.
You found a plane once, Ben?
What?
Ben once found a plane.
Ben once found a plane.
Or maybe more than once, I don't know. Yeah,
Wow.
there are
So I find
sometimes-
5 urchins strung together and he finds an airplane. Nice job, Ben. Where was it?
Bombs.
Bahamas. Do you want to tell us something about it?
I'll take a look at this.
What about you, Adam?
I'll take a
What's
look
something
at
you
this
didn't expect to
Coral
see?
over here, Ben.
Expect to see.
Yeah,
What's something you
you
didn't you've seen on the seafloor that you never expected to see?
We found a bulletproof vest that one time. It was interesting.
Helmet. Helmet too.
Helmets and bulletproof vests, planes. What about you, Alexis?
I don't know.
So far in this grand scheme of things, my 5 urchins strung together are on the very bottom of cool.
Oh, I think we have a Victor Gorgia.
I think that is a Victor Gorgia.
I just love
last time we were
I'll just
here,
come
we
in
had the skiing
a little
fish.
bit.
Remember that, Randy?
What'd you say?
Last time we were in Pitta, we had the skiing fish.
Yes.
I have video of it.
Really?
Yeah.
I would love to. I haven't thought about that in a long time.
What's a skiing fish? Why
Yeah,
would I
yeah. It
remember
was
that?
sort of.
Yeah, me neither.
It was sort of sloping down.
Let me turn on the side.
Oh, wait,
Okay.
I didn't really see where I meant that.
I'll come out for you.
All right. What is that? Let's see.
Someone asks who gets to name the unknown creature, so like new species, and often that it requires a taxonomist, so someone that focuses on that particular group of animals. And then every once in a while things are named after certain individuals, but for the most part the scientific names come from characteristics about the animal. So something about the way its morphology looks or where it came from.
This one is obviously Victor Gorgia Alexis Weinigus.
I think we already have a species name for this.
I'm just.
I know.
Sorry. Yes, exactly. He was going down, down, down. Part of that
Oh,
made it
Randy,
into
you
one
got
of
a
the videos.
snail.
Did it?
Yeah.
Randy, you got a snail.
Oh, hang on. Snail. You don't remember that fish?
Well, I have to
All right,
look
let's
at
take a
it.
look. Victor Gorgia. We don't have a Victor Gorgia button.
Oh, yes, I was told to do that and I did.
And then this is an astroschema, and then we have. Is this a corallivorous snail? Probably.
Maybe.
It looks like there's some damage at the base. You see?
Let's see.
Maybe. I can't really tell actually. Mm. We also do not have a snail button.
As an associate?
Mhm.
Okay.
All right, I will. Hang on, let me give you some button.
Are you good on this, Randy?
Yes, I am.
Oh, there's a gastropod button.
Oh, okay. I just didn't see it. Thank
I
you.
use the scientific name.
I was literally looking.
Okay, that's good. Thank you.
Gastropodensis. Just kidding. Yes, I should have looked for gastropod.
It did sound kind of funny when I found it. I had to call it topside. Hey, topside, there's a plane down there.
Yeah.
Plane.
Was it on what ship were you on?
Stuart Johnson.
Oh, okay. Was it the Johnson Sealand? Okay. That's cool. That's even cooler. I feel like.
Uh-huh.
Found it.
Oh.
So when he found the plane, it was in the Johnson Sea Link, which is a sub.
Oh, that is cool. So you're
that's
actually
now
in it.
decommissioned, Yeah.
Found the ship too.
Yeah. I never. That was before my time, unfortunately.
Found the ship. It's never been seen.
Oh, cool.
It's almost intact.
We found shipwrecks in shallow waters in the Phoenix Islands. So you just follow the black scrubby algae because it leaves as iron.
Oh.
The iron in the ship degrades and leaves this fuels this microbial cascade that uses the iron, and so you get this it's called the black reef. It's a really clear and distinctive consortium of bacteria combined, like iron oxide or iron utilizing bacteria mixed with an algae. And if you follow this black reef up current, you'll almost always find the iron that it came from.
Oh,
Yeah.
wow.
Found a schooner in the Great Lakes.
Oh, that's cool.
And they had
That's
a
one-
lady in the bow and everything.
Oh, that's cool.
Yeah, it's so beautiful. It's almost intact, but I'm sure within a year of publishing the location of that thing,
Oh,
it's
yeah.
shallow water. It's just, it was never seen before. There wasn't any charts.
Did you identify the wreck?
They did, yeah. Because we were scrubbing the name.
What was it?
Can't remember now. It's in the books.
Dave, you are very welcome.
It's one of your fellow Canadians published it. Joe McGinnis. Joe McGinnis.
Pull the camera up a little bit.
Yeah, that's another urchin.
getting close to 2,000 meters now. Working our way up to hopefully 1,400 during this dive. That is a good, Jason, the thrill of discovery is addictive. It really is. Like, we talk about kind of like the sleep deprivation side of it, and some of that is because you don't want to stop watching even when you're not on watch.
Mm-hmm.
So you sit in the library or in the mess, and the dive is always streaming throughout the ship, and so you're kind of just always waiting for that next thing around the corner or up the knoll, you know. It is really exciting when you come upon something that you weren't expecting. Yesterday, that big wall that we were on at the end of the dive was kind of one of those situations where you had an inkling that something was going to be great at the end, but when you finally get there, it is really a thrill.
Every time. And things can change like that. You know,
Yeah.
this could go
Yeah.
from rocks and sand to nonstop corals in an instant, or it could turn into, it could stay rocks and sand. It could, I mean, you never know.
I'm just gonna pan left and see.
We could
what
see
this
some
is.
incredible creatures swim right by. We've had a couple of incredible creatures this trip. I personally like some of the octopuses actually that
Yeah.
we've seen, but there have also
Looks
been
like-
the charismatic megafauna, which everybody else is really into. Like,
Victor Gorgia again.
yeah, I got it, and a sponge. Let's see.
Great.
Yeah.
Yeah, somebody asked, what's the forest of coral at the end of last night's dive? We're good. We already, we just looked at one of those.
Yes, there was a forest at the end of last night's dive, which we're going to try to get back to because it was, all of us were, I don't know, it was burned in my mind as like,
Oh,
we got to go
I've
check
never
this out.
seen that kind of density of the same species in that kind of
Yeah.
arrangement.
So we're trying to figure out how to get back on this trip, so I think we can. But it all depends on timing and currents and winds.
Yeah.
I'm gonna go grab breakfast and I'll come back in the loop. We got plenty of
Perfect.
Those that depth of between like 1,000 and 300 is really interesting to me. I am a big scleractinian fan, so those hard corals are what I've spent a lot of time focusing on. So whenever we start to see rubble and that kind of evidence. Oh, can we take a look at this, Ben, right here?
Did you see what I tweeted about you last night?
Yeah, I did.
Alexis is obsessed. I sort of outed your obsession. Not that it's a secret.
It's not a bad obsession.
No, not even a little bit.
Yeah, I always get really excited about trying to find the live colonies that lead to that rubble. Oh, there's a big whip over there.
Yeah. See, the community's changing. We said just around the corner we might get more corals. If we just said just around the corner we might find a pirate's treasure chest filled with, you know.
Doubloons.
What were we thinking? Man, we were thinking we should find the corals.
I have been on one of the cruises I did with the Okeanos and the Atlantic. We had a target for what could have potentially been a shipwreck, and when we got there it was just a pile of rocks. But that happens a lot. Oh, sorry. Yeah. It's interesting because there's a lot of shipwrecks that are just undiscovered, essentially. People know that ships went down, but they just don't know the exact location. And so when you think about how we're just kind of down here with a flashlight, it can be really hard to locate something, even though somebody knows the lat and long of where that ship went down. That doesn't mean that's where it is on the seafloor because of the currents, and over time things can shift around.
And even as it sinks, it moves. It doesn't
Right.
sink
That's what I
exactly
mean.
down. Yeah, yeah. And then there's often a debris field, and it gets covered up. And
I like that. Pirate chest filled with corals. Yes, please.
there you go. That's a good idea.
It's a very small colony with a small ophroid.
What are you? You look like. All right. I think this is. Can you?
Yeah.
Can I just want to try it before you tell me what it is.
Okay.
I don't know. I'd probably just say Plexaurid.
Yeah, that's what I would say. It's hard when they're this small, but the way that the polyps aren't like coated in sclerites and there's not any spines coming out of the retracted polyps makes me lean towards Plexaurid
Yeah.
with a not very small, but appropriately sized ophiocanthid or astroschema. Sorry.
Yep. Got it. So yeah, the down here with the flashlight line is really from Jan Whitting, who says it's like, we hike up these mountains on a teeny tiny hiking trail at night with nothing but a flashlight.
There was
And
a
it's
whip
such an
coral somewhere
appropriate
up here.
analogy for what we do.
I don't know
And there's a
where.
little star
Okay.
up there.
I don't know where the coral went.
We find things unexpectedly too in weird ways in the shallows. I started
Yeah.
diving sometimes.
Yeah.
So,
Yeah. Oh, here it is, right here.
When I'm
Sorry.
on
We'll go look at that.
and then we should look at those
Okay.
too. When I'm on scuba sometimes, especially when I'm like my head's in the corals, I've started putting a GoPro on my back to see what
Yeah,
what
yeah.
passes over me. Because when you're really focused, it just looks like a cloud, but sometimes it's
Yeah.
crazy stuff that just swims over the back of you.
So yeah, your friend has that saying, but I think that's also
Yeah.
like kind of just a widely used
Yeah,
analogy
exactly.
because it is something that is the best way to
Describe what we're doing.
think about that.
I agree. Is that a bamboo?
I think so.
Whoa. What are you? So we're getting a question about temperature change, and we've gone from a freezing cold 2.1 degrees C to a balmy 2.2 degrees C. So I don't think it's been a very extreme temperature change, but every slight bit might change or influence. I think, but we have climbed a couple, quite a little bit in elevation here, and so
Yeah.
I think that we might be seeing a change in depth and or substrate.
Yeah.
So there's a predatory tunicate in the back.
Oh, good eye.
And we need to
We'll
turn
settle
that.
down here before
Yeah.
we zoom in.
Oh, now we're back down to 2.1 degrees C. So I don't think there's been a temperature difference, but we've certainly seen a depth difference.
Mm-hmm. And that's what Steve, who's on board, has
Right.
done a lot of like distribution work.
Actually, you guys should check out his blog. It just got posted.
Oh.
And he wrote a beautiful blog about all the things that we've been thinking about in 2017 and what we findings from the Phoenix Islands archipelago so far. So if you haven't checked out the most recent blog related to our cruise, this one is by Steve Askovich, and it is the Schmidt Ocean Institute webpage associated with this cruise. You can check out his blog.
Can we either ISO down or IS down? Great, that's so much better.
Bamboo coral
Yes. Little
with
mice.
a little shrimp. Ding. We're really lucky to have Eric Olson on board. He's an absolutely stellar journalist, photographer, and videographer, and he has taken some amazing photos. I haven't actually checked out our cruise website recently, but I think some of them are up there as well. And so if you look at the gallery, you should be able to see some posts of our dives straight from Sebastian and some of Eric's work too, who he's really phenomenal. I think there's also a blog by Constance Sartor, who is our artist in residence. So she's an artist on board who's doing some incredible work with upcycling magazines in order to, and it's such fine detailed work. It almost looks like paint, but it's this beautiful, with the urchin too.
Isn't some of it paint? Doesn't she paint as well as do
Yeah,
the
but-
collages?
I think so, but I
Okay.
mean, the collages themselves kind of fade right into the paint. I think they look gorgeous. So she's done some beautiful work, but she's also a scientist. She's finishing up her master's at the University of Guam, working at shallow water transplant experiments. That's the word, and looking at the transcriptomic
Okay.
responses to transplantation. So we have really, besides the science team, we have quite a lot of
Okay, we can carry on.
talent in art and journalism, photography, and videography, which is lovely.
Yeah, that was a why can't I say this? Predatory
Predatory.
tunicate. So they are kind of like underwater Venus flytraps, where they will just snap on something if it comes into their path.
You know, this is just random chatter, but I teach evolutionary ecology at Boston University, and every year I give my students at the end of the very last lecture of the semester is what I call the Your Choice lecture. So it's the one thing that you wish we had had a chance to talk about that we haven't had a chance to talk about, right? And so students vote, and I pick my favorite topic of the things that they're asking for, and I put an ad hoc lecture together on the fly, different every year. And this
my
year
gosh.
was on carnivorous plant ecology,
Oh.
and it was so fun.
That is fun.
It was so fun
How
to
many
think
different
about.
carnivorous plants are there?
Oh, there are a couple
lot?
hundred species.
Really?
Which is amazing. And so we
Wow.
went through sort of like sundews and bladderworts and fly traps and pitcher plants, and it was a huge variety, and went through the ecology of how and why they do what they do. It's really one of the things I look forward to every year, which is seeing what they come up with and putting something random together.
Alright, let's go.
Me.
Okay.
And you can answer that question that came in. Because you are more equipped to answer that.
Water counter above you. Okay.
I'll take my cup.
All right.
I'm sorry, Randy, I just
That's
jumped
good.
at that.
All good. I'm not hungry. I'm just tired.
Okay.
But I do have to eat before 7:30, so come send somebody in.
Yes, I will.
Thanks.
Or I'll come back.
Okay.
Oh, your meeting's at 7:30?
Yeah, I'll be fine.
Okay.
All right, so the question, let's see, is do you have the ability to monitor ocean currents within the water column directly above you? The answer is not really. So the ship has a couple acoustic Doppler current profilers that, depending on the frequency, penetrate different depths, but generally not this deep. So the 75 kilohertz system the ship has will do 600-700 meters. The 300 kilohertz system only does a couple hundred meters. So, where I was actually just talking to some of the ROV engineers the other day about how nice it would be to be able to directly measure the currents from the vehicles, and there's a couple potential ways of kind of hacking some of the ship's navigational, or the vehicle's navigational sonars to potentially get it, and that's something that'd be pretty high on my wish list of things to do. So they have a bottom tracking system called a Doppler velocity log, and you theoretically could back out some of the influences of the currents using the inertial navigation system and the sonars on the vehicle. But I don't know I've never seen that done successfully, but in theory it's possible.
Cool. Thanks, Brian. Hey, Eric.
Hello.
I'm just talking about you.
I'm back.
You want to hang up here with Brian?
I'm sorry.
You want to hang out up here with Brian?
Sure.
You can log, right? Well, my
Yeah,
computer.
sure. Just
Log.
give me my computer.
Boy,
Not missing enough
that's a
time.
beast.
Thank you.
Is it okay?
Yeah.
Great. I'm going to sign off and leave you in Brian and Eric's very capable hands. And see you later.
Hopes I haven't missed anything.
Looks like a little, probably Cusceolophidiid there.
You didn't pick them up. You did pick them up?
It's trash.
That's very dangerous. I wouldn't think
you to do that. So we're currently just crossed 2,000 meters. We're now 1,992 meters, coming up on a deeper dive on Howland Island in the Pacific Rhode Islands Marine National Monument. So far this dive has been a little bit sparse on life. We were hoping for a little more density of coral and sponges, but understanding the areas they're not is every bit as important as understanding the areas they are, as we're thinking about trying to understand what controls the distribution of these animals and where they are. Ben, can we go take a quick look at that coral coming in on the left? There. I don't think we need to set down. If I just let me know when I can kind of do a snap zoom. All right. We'll just take a quick look on this to get a better ID. This is probably a type of acanthogorgid or okay, I'll hold frame and let you fly it. Acanthogorgid or plexaurid. Yep, probably an acanthogorgia, but coral ID is not my expertise. Let's take a quick shot. All right, I think that's good enough for them to get an ID. Well, if I can maybe do it focus. There we go. All right, I think that's probably good enough. We can move on. Coming full wide. Can we take a quick look on that lip right there?
Small one.
The big one. Small one, I'm pretty sure is a hemicrallium. Can't tell, this might be, not sure what that is. You stable? Okay. This is probably some kind of bamboo coral. But I don't see the nodes here, but that's probably because it's just a little on the fleshy side. See if I can see a spot here with actually the nodes in it.
Strange that it's on one side, right?
A lot of the deep sea corals, you will see that, and as a, they tend to have their polyps on the down current side and the turbulence, and they try and kind of pick particles out of the turbulence. Now this is a benthic ctenophore living on the corals. These are actually one of my favorite coral associates. So when you think about ctenophores, these are normally comb jellies that you think about being free swimming in the water column, but a group of them have evolved to sit on the seafloor or attached to a coral, and then this long tentacle you see out here to the right as their feeding tentacles. They have two. and they just kind of sit here and hang out on the coral, use the coral's protection, and then catch food out on these long tentacles.
Do they float through the water and then embed, or how do they latch? How do they get up there and latch onto that spot?
I don't know much about their life history, honestly. My guess is that in a younger larval form they embed, and they probably pretty much stay where they are for the most of their life once they stick. But I don't think we really know the answer to that, but that's the assumption I would operate under. We got need there. All right, let's keep going. So as we're moving along, you'll see those two green dots floating around the center of the image. Those are our lasers, and they're perfectly parallel, so that allows us to measure how far something is, no matter how far away it is. So they're 10 centimeters apart, and since they're parallel, that doesn't change. So as long as we've got looking at an object that's pretty much flat on to the perspective of the camera, we can use that as a scale to get a sense of the size of the animals. Like we're coming up on a glass sponge and coral that I can't quite tell what it is yet. Given the color, I'm kind of guessing it might, well, maybe not. Mind if I just snap zoom on that? Yep, that's a Victorgorgia with a glass sponge behind it. Maybe that's all we need there.
Yeah, go ahead, Mike.
So the wind is still pretty low at the moment. If it does change, I would expect to drag my head in to starboard a little bit.
Okay.
Shouldn't be a problem for you guys, but just to let you know any dramas will update you.
Okay, sounds good, Tony. We'll just slide it behind you here.
Yeah, it should be fine for now, but yeah. You can see me, if you see my head swinging, it's because the wind is shifting direction.
Okay, copy that. Thanks.
So as we're moving along here, it's good to remember that the ROV is, currently almost 2,000 meters under the ship. That's a little over 6,000 feet, and we're tethered to the ship by a very thick armored cable, and so this is a single-bodied ROV system. If you're used to watching Okeanos Explorer or Nautilus, they use dual-body systems. There's two vehicles tethered to each other down on the seafloor. Here, there's just Sebastian, and they use a series of floats along the heavy cable to give it some buoyancy and make it close to neutrally buoyant. That's how the vehicle gets its mobility at the seafloor and isn't dragging around a super heavy cable the whole time or being pinned directly underneath the ship. The single body system really does give you a little bit more flexibility in terms of moving around than the dual body systems do.
okay for a few more minutes, Brent?
Yeah. I've already eaten, so
Oh, yeah.
yeah.
Does the spool of the cable on the ship both go out and come in as the ROV creates or
Well, I'm gonna throw it to these
diminishes
guys. You guys
slack?
wanna answer that?
No, we manually pick it up and pay it out. Like right now, I'm picking it up as we go.
You're picking it up right now?
Yeah, right here. It'll
Okay.
lever.
And you're following that delta number. That's how you know what the sort of curve is. And there's a
Yep. Yeah, so when it comes off of the shoe wheel on the bottom, goes straight down, then hits the 10 floats they put on.
yeah.
So it comes down, and then it'll go up, and then it'll drop back down, and the tube we put on, and then it'll pick back up and then follow down to the ROV.
Oh, really?
Yeah. So it kind of goes like this.
Oh.
It's called the catenary. It's like our buffer between the top of the vessel and the ROV.
It's called the what?
It's kind of like it's called the catenary.
Catenary. That's that shape.
Yeah, that gives us like a buffer, so when the ship heaves, it's not yanking us up and down.
Oh yeah. It's got a lot of wave sign slack in it, right?
Yeah.
Oh yeah.
Yeah, if we were to just
So
go
this is-
straight tight every time the ship moved, we would feel it at the bottom.
So this is a crinoid or feather star. You see these pretty commonly, but a lot of times we just generally drive right by them because they don't have the same level of they aren't ecosystem engineers the same way we consider corals or sponges, but they're really ancient species. You see these non-stalked crinoids, comatula crinoids, in the shallows as well. In the deep sea, we also get stalked versions that we'll hopefully find a little bit later because I think they're really beautiful. They're commonly, these are called feather stars, and the stalked versions are known as sea lilies sometimes. All right, we're good here.
Because that was because that's what triple index went from 80 to 74. Try it right now. Yeah, no, there's a difference in like 6 meters. And then Caro, what did I have? I'm pretty sure INS is lower.
So I'm sitting here with Eric Olson, our multimedia consultant, correspondent. What's the C stand for? MMC? Multimedia?
Correspondent.
Correspondent.
I think.
Okay.
Yeah.
You want to introduce yourself and tell you a little bit like what you're doing on the ship?
Yeah, I'm Eric Olson. I'm multimedia correspondent, so I'm not only documenting what's happening on the cruise to some extent, I'm doing a lot of filming in video, as well as still photographs, not just of, what's on the screen here in front of us and what you could likely see on the screen, but of what people are doing sort of behind the scenes. Doing some interviews with people to talk about what their research is about, what their interests are, what the goals of the cruise are. Some of those, will be packaged into ideas. Some of that video will be packaged into ideas, small videos that tell a story about this particular cruise and the goals and whether we achieved them. First one's out. It's on YouTube and I think it's available off of the Facebook page and SmithOcean website. That one, the first one is a discussion with Randy Rosen and Tim Schenk, largely about areas beyond national jurisdiction. So ABNJs, which is not where we're in now. Now we're in an EEZ zone of the United States. But the ABNJs are sort of the vast open ocean, the open seas that aren't the province or jurisdiction of any particular country. And it turns out that that's actually most of the open ocean. And the question is, there's so much vast, amazing life in these areas. No one's really responsible for protecting it. So the question is, are we at a point with the emergence of deep sea mining and other extractive industries, where there's the potential to go down and exploit these areas for resources? Maybe we should know a little bit about them first. So that's sort of one of the goals of the cruise, as I understand it, and so that's that video tells that story through the experience of Tim Shank in Randy Rojan. So yeah, that's what I'm here doing, taking a lot of photographs, a lot of video, and more than anything, to be honest, enjoying this experience of living at sea for 34 days and, you know, gaping in awe at the wonder of the deep ocean, seeing things that nobody's ever seen before, literally, and hanging out with people who are a lot smarter than I am and asking really stupid questions and getting really satisfying answers. So it's pretty good job.
Well, we really appreciate you being out here because no matter how all the good science in the world doesn't turn into good policy unless people know about the science. So that outreach and communication is super valuable. If we just were doing this in a vacuum, it would be worthless. And so that connection to what we're doing from the science side turning into awareness, education, and policy is critical. And it really makes what we do, gives it value. So we really appreciate your help in that, Eric. Thanks.
You bet. With that, I gotta go to the meeting.
All right, enjoy.
So that's just the difference between this control panel and I've noticed a couple of weird things like pumps, you got two pumps on the bottom side. But the other ones you just turn them.
Oh, really? Yeah, it used to be.
Leave it up, you can press them, but you didn't have to press them.
Oh,
They're
okay.
on.
How far are we from waypoint 4?
What's your
36 feet.
four?
36 feet.
360 meters?
240.
It's a good estimate,
I
wasn't
was
it?
just thinking about doing a transect so we get a little quantitative data. Can you drop us a point 100 meters out from the current vehicle position? Perfect.
You happy with this height?
Yeah, this height is really good, so we'll just, you can even pick it up just a touch. So if we want to just basically head that direction, we won't change our heading much, and kind of just go straight along that, 0.2 of a knot, something like that over the ground, and then I'll get us a link. So for those of you watching at home, we're actually going to start a video transect here, and so we're going to run 100 meters, more or less in a straight line, keeping an even altitude, not stopping and zooming on things really. And this will give us a better sense of being able to do some quantitative analysis of what the abundance of organisms are here. And so we'll just kind of fly over the seafloor, about half a meter above the bottom at a nice measured pace. And that'll allow us to, like I said, do the quantitative, do a little more math to this than having us wandering side to side, stopping and zooming, so we can actually calculate the number of organisms per unit area.
Do you want me to log?
Doesn't matter. So, seeing questions about the video, the native resolution of the video is 4K, but I think we believe we are streaming at either 1080 or 720, just to reduce the bit rate a bit to allow it to go over the satellite in real time. So after the cruise, when we can do hard drive transfers, the YouTube video will be upgraded to the 4K resolution that we're seeing and that we'll do our science on. But that's just too much bandwidth to push over a satellite in real time, given the current state of telecommunications and ships and the expense of satellite time.
Which camera's frozen? How is that possible? Because somebody doesn't have it on the right camera.
Yeah, that's the delayed one, isn't it?
That is. You didn't push the button that says RV Winch.
Oh, there's the small.
Oh, yeah, I saw that.
It's raining.
No, it's not.
Looks like it.
It is raining.
It does look like it's raining.
Oh yeah.
The
How's
fun
this- does anybody know how the sunrise was this morning?
No idea.
I walked out.
I was here with you.
I walked out about 6. It was nice out. Big storm and
Ooh, storm a brewing.
Yeah, it sounds like it's going to be wet and drizzly for the next few days, probably. Got some type of stalked coral coming up here. So it might have some hydroids and a crinoid on it. Probably a bamboo, I'm guessing.
Yeah, looks like.
You talking about telepresence, Brian?
I was a little bit, yeah.
When I walked in, what were you saying? Anything cool?
Just talking about video resolution.
Oh, why it was crappy the last 2 days.
Oh, I actually didn't mention that. No, I was just talking about the 4K versus streamed rates.
Oh yeah, YouTube won't let us.
Are you still restreaming through Zoom?
YouTube.
That,
Huh?
No.
are you
No,
What
it fixed. Omni routing.
It was done.
So,
It was routing.
so
Yeah,
now I can
Omni
finish
access.
watching the YouTube video that I was watching the other day?
Yeah, they fixed something, updated something 3 days ago. That
Right in the
broke,
middle
and
of
then
it,
they
and
kept
all of a
telling
sudden,
Toter it wasn't routing,
the
and
whole
then
system
sure
went
enough,
down.
it was routing.
Is it cephalopod week?
Well, sorted. Look at that, flat as can be.
So it wasn't on our end at all.
It wasn't on right now.
Oh, the Zoom was a good workout there. Good job.
Zoom was a good workaround. It was better than nothing.
Oh, you jinxed it. Now you got a spike.
And it doesn't. It's better than the seesaw that usually happens.
Yeah.
What's a spike mean?
I know that people didn't like the Zoom, but it was either that or nothing.
So you just shot the stream through Zoom, and then they picked it up on Zoom and put it to Wirecast?
So on Zoom webinar, you can stream to a custom RTMP. So yesterday, the first day was just like I was standing on the aft deck and I had this epiphany of like, Zoom webinar can send it to YouTube. So we just did it straight to YouTube from Zoom webinar. Yesterday, I had Hannah set it up to restream. So then we were able to send restream to YouTube and Facebook at the same time, which is how we normally do it from here. It's just Wirecast. But that RTMP message, whatever it was, they had
Do
routed
we have an update?
weirdly or had blocked.
That's really loud. Hey, Coop. Coop, I think you have to say hi to everybody.
Yeah,
said that to me. She said it looks like a big sea pen.
Aw.
But just to me. I was like, how's she ready to get a private message?
Well, don't let people creep you like that.
Well,
It's a big urchin sitting there.
There's a coral coming up.
And here comes a coral. That is a big urchin.
On watch, we currently have. I'm Alexis. We have Brian. We have Adam. We have Chris. And we have Deb.
I just thought about it. And I was like,
Henny?
Yeah, I think so. It's hard to tell, but
Yeah, I know, but
okay. Sure, why not? We'll just, as long as everybody's sitting over there with
it looks like we're about halfway through the transect. We're approximately 50 meters or so through here.
So I would
And
say,
we'll continue on for another 50 meters.
probably
And then we'll kind of resume our normal looking around.
what we put in for it.
big tube anemone or anemone.
Alexa, Brian, Adam, yeah. Oh, no, I forgot Chris.
What's today? Friday. Well, it's 17.5 hours. That's what I mean. And then that's 17.5
Little
hours.
stocked crinoid.
2 hours. No, but you could skip seamount 4. I don't actually think that's gonna be, I mean, unless somebody's like really super needs it, I think it's gonna be pretty deep. But I don't know if it's part of, like a need in a particular latitude or.
We're getting close to 1900 meters.
I think seamount 3
So we
is
started
more
this dive at about,
interesting for
let me see,
the data.
2400 meters, and we're hoping to make our way up to 1400, so covering about 1000 meters in depth during this dive. We started at roughly 7 hours ago, and we will be continuing for at least a couple more hours today. What's the date? Is today the 19th, the 18th?
18th, ship time.
Yeah, ship time.
Still 18th, GMT too. Oh, there's a little Crassagorgia.
Oh,
That's one of the first ones we've
Crassagorgia
seen.
is the first one.
So now we're coming into a much steeper terrain here.
How are you gauging the length of the transect?
Drop near point on the
Oh, gotcha.
maps. I'm just that good. I can, tell how hard the vehicle's been just by watching the seafloor.
Okay. Mr. Smarty Pants. Got people viewing from all over the world. We have people earlier were viewing from Scotland and Switzerland, Sweden and the Netherlands, people from the US. Hopefully some viewers from Pacific Islands.
So we've got a sponge and some type of stalked coral over here.
Mm-hmm. greetings from Canada. Hello, California. When we talk about sponges, they are often, that's some type of little chrysogorgia, right?
Yeah, it looks like Chrysogorgia anthomastus.
These are sponges that are, a lot of them are what we call glass sponges, so they're made of silica. So they are not the kind of sponge that you would use on your skin, or you would have lots of little prickly bits stuck in your skin. Demo sponges is another class of sponge that is more of the, we find them in the deep, but also the shallow areas is where you would collect sponges, natural sponges that people use in their showers and things. Hello from Seattle, more people from Canada. This is a little local high maybe, or is it just, are we approaching a local high, or is it just a more narrow?
I think it's just we're gotten up on a little narrow ridge.
Okay. Sea star. I have to find a time to fix the buttons in between dives.
It's funny, in my deep sea career, we use enough different loggers, and every ship has a different kind of logging system. I feel like the amount of time I have spent talking about what buttons we wanted available to push to log things has been a lot. I don't know, I have no idea to quantify how much, but I'm surprised how much time I've spent discussing buttons.
Days, years.
Literally days. I mean, I think at one point when I worked for NOAA, we spent a week with our data management team discussing, at times arguing, sample naming conventions. And it was 5 or 6 people for an entire week discussing how we were going to name and categorize the different samples we collected.
It's like I would say Corral a day from here, but that's
I would
pretty
agree.
far out.
That's fine.
Ooh, we got our first
Iridogorgia.
Iridogorgia. Hi, sir. Good morning. An Iridogorgia. We're in the middle of a transect.
Okay, how long's our transect?
We are
We're doing 100 meters.
we are approaching the end.
We are approaching the end of our transect.
Nicely done.
This is becoming much more narrow over the last 50 meters or so.
And we've seen a marked increase in, I mean, it's still sparse, but a pretty marked increase in corals.
That's good.
That was the first Iridogorgia we've seen all dive.
And we just picked up the first Chrysogorgias at all
Or we didn't
10
pick it
minutes
up.
ago.
We saw
oh,
it.
sorry.
Okay.
Yes, picking up in the environment, not picked up as in collected. What have you collected? Anything?
Barely. Where's the? It's under your computer.
You've been seeing that. That's that.
We've seen that.
Corral.
Yeah. Which, if you are interested in a sample, you're more than welcome. I think that's the Paragorgia with the.
That is.
It has those zoanthids on it.
Okay.
So I don't know. Without zooming, it's really hard to name those. That is.
Basically started the
Perfect. All right, let's say we have completed the transect.
249 was. Yeah, it looks like
Yeah.
it. I'm sorry, I stole the pen.
All right, so 1851, and we're done with the transect. Tim, you want to take over?
I'd love to.
All you.
Can I use your computer to log until
course
he's
you can.
until
Do you mind Alexa staying a little bit?
I will stay a little bit.
Oh, well, you can go. I can log.
No.
We'll just run transacts. Right? Hi, everybody. This is Tim Shank. You're on the overnight.
You're on the
On the morning.
now early now
Somebody's-
morning. I was
somebody's
on
night.
the overnight.
Right.
Hi everybody.
Hi, everybody. Shake hand.
You gotta keep Slack open.
We got,
you know.
Slack's
I
got
mean,
to
you
get
don't
up. We
have
should
to-
have a. I mean, this is just. We don't have one for the.
Like,
What
you
dive
wanna
is it?
zoom on that?
What dive is
We do have one.
it? You ready to zoom? Just a pair of gorgeous. You want to zoom?
It's up for yeah, sure.
Can
Alright.
we just do a quick look here? Yeah, sure. Let me get warmed up here. All right. Is it? This is dive number.
425.
Yeah. So we don't have a channel.
Yeah, we do.
See?
It's
This
way
is
up
how it's-
here. 425.
Okay. See, there it is. Okay. Oh, we're ready then.
Because I have layers that are.
Do we do this and you do that?
Yep.
Thank you.
So it's in the Jebco, but I don't have the files that are just that.
Right.
So I'm trying to figure out whether we actually have more data.
This will be fun.
I have this.
Paragorgia with zoanthus.
Yeah, that's what I think.
Astro scheming. So anybody else? This is what you've been seeing, yeah?
Yeah, or we've seen a couple, not a ton of them,
It's a
but
little bit of a cower style down here on the bottom or something. Or is there a Umunita going in? It's a Umunita. Awesome. One back behind it too.
do you do
Okay.
Unity?
Keep rolling up. It's great. Okay, so what happened here? 1887 meters. We started at what depth?
24.
17 hours. So we also want to get to my job, we want to do a lot of.
2400.
Obviously not going to the shallowest to be that obviously not the best weather windows.
Keep this open. What's going on?
So, best weather days Tuesday, I don't think we're gonna time.
There's a little Chrysogorgia.
And that could.
You've been seeing those too?
That was the second one, so we hadn't seen those until very recently.
It's nice to keep some overlap like this. It's cool. What's our sonar say? We got stuff way out ahead of us still?
What's that, sir?
Just looking at the sonar. Can you explain it to me? This little low relief we're having?
Yeah,
Is it gonna,
it's
has
gonna-
it been undulating or has it been?
Yeah, it's been this really weird, very
Yeah, just
rounded
kinda like-
ridge, not jagged at all. Almost like
So I think this,
When I was watching, I saw more of a slope with sediments and
Yes, it was very sedimented before, and we don't really have
sort
the
of lost the
sediment
sediments.
anymore.
Yeah.
so that's off the table. So. Do you have the protected area boundaries?
So there's a question about
Yeah.
if we're here. You can use the pen. You're
Brilliant.
not a pencil person.
Not like this.
Um, There's a question about if we're on the ROV. We're all on the ship.
Okay. Trick pin?
Oh my
What's a
gosh,
trick pin?
you-
Here we go.
We're all on the ship. The ROV is exploring under the sea floor, under the surface, and so it's called a remotely operated vehicle because we operate it remotely. And so this is not a manned submersible, which would be what we would be if there was somebody in it, you would call it a manned submersible. But so we're all sitting in a control room on the ship. We have 2 ROV pilots who are flying and using the manipulator arm, and then we have 2 scientists here. My name's Alexis, and we have Tim, and we have a marine tech who is actually working on other things right now,
Yeah, I think they're dive planning for tomorrow's dive.
or this afternoon's dive.
Yeah, I don't know what day it is. There's something stopped coming up here.
Do that one, just to get our latitude.
Looks like, oh, we got a big base right there.
That's a massive base.
Massive.
Well,
Looks like
what
maybe
I've
a
seen. Is that another-
hemicorallium.
Got to tilt up a little bit, maybe.
Yeah.
You can take a look at what it is. What is that? You got that over there?
I think that's hemicorallium. Oh, I'm supposed to be along. Ooh, nice big bamboo coral.
Tim, introduce yourself.
Yes, I will. Can we just take a look at this?
Yeah.
Introduce myself? Yeah. Hi, I'm Tim Shank. I'm a deep sea biologist from the Woods Hole Oceanographic Institution. My interest research on this cruise is looking at the biodiversity of animal life that lives on the corals and sponges. Looking for patterns of association and fidelity. These animals you see on the corals are often partners, or can be partners for life, and they're very specific. So certain coral species will have a certain associated fauna with it,
You
like
see
a
any
worm or a
associates on
crab
here?
or an ophiuroid. I see something over here that's striking my eye. You look for color differences and then morphological differences. What is that? It's, I would ask if I could zoom. Is that okay, Adam?
Yeah, go right ahead.
That's where we want to wrap back up.
See, some of the polyps will be modified a little bit, you know,
Yeah.
or
Just
discolored.
staying on
I was focusing on
There's-
that right there. I'm not sure what that is. It's-
Oh, it looks like
see
a
it behind it.
ctenophore. Look at
I see
the.
a strands coming off of it, yeah.
Yeah.
But that, it runs
It's
the
a
back,
lot
it
of
looks
it,
like.
yeah.
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 Big benthic ctenophore here that got caught in the...
No, Steve was on earlier this morning, like
What
4
is this?
hours ago.
Yeah.
Yeah, that was Brian.
I totally missed it. Okay.
It's
Do you see
too
anything
bad
else
that we
here?
can't see the other side of it.
Do we want to sample it?
That's up to you. It would be yeah.
Do we
I
need
mean, you
this?
could take that branch if you wanted to. Right below where you start to see it.
What do you mean? I don't want the, I don't need the ctenophore.
Oh well, then no. I don't
Do you
think
want
we
to
need.
sample this over here or something?
No.
Okay, all right. Anything else you want to see here? Are the lasers on?
Check.
You could say here.
Yes, they are.
Okay, great. We'll go down to the bottom. One more little zoom here. Is this your first time you've seen this on the dive?
Yeah. Heard.
I normally am
We've
able
seen
to watch
small
the dive and
bamboos,
keep
but-
up. See, there's like little galls down there, maybe some, or is that?
Yeah, those are looks like
polyps
this.
growing on
Well, it
the-
looks like the skeleton has overgrown something.
Yes.
So it's
Yeah, I wouldn't call it a gall. Sorry, that's
more
not
than
right.
yeah.
All right, let's go down and look at the base here for a second. Just get some lasers on the base.
It is a very thick base.
Very
And this
thick
is like you get this this over calcification of the nodes, so you
Yeah.
can't see the nodes anymore.
It's big.
Yeah, I mean the stem is 10 centimeters.
Here on the river, you look
8
It's pretty
centimeters
amazing.
maybe.
That's a monster.
involved in diving there and you're going in where you
Okay.
want. It's just a question of
Curious. There's no ophiuroids on this. No little stars.
Mm-mm.
I would
No
assume
crabs
so. Even
either.
around the base. Slim pickings.
Slim pickings.
Okay, Adam, thank you. Did you log that as a?
no sample.
Locked it
No
as a
samples.
coral.
Yeah, we'll go. I'm just gonna square the camera up a little bit. Is that better? If that helps you. Oh, let me just. I'll come down and look at the basket then. Yeah, we're centered. Chrysogorgia down below. All right. Onward and upward. What we're diving so deeply today is to continue a transect, if you will, of depth coverage around Baker Island. Yesterday we did a more shallower dive. That went up to shallower than 400 meters, maybe 360 meters or
Yeah.
so. And
Started at about 900.
you've seen this yet?
Yeah, I think that's the Paragorgia.
Just snap zoom is all I'm going to do here.
Yeah, it's a Paragorgia.
No zoanthids though.
No zoanthids. Astroschema.
Sorry, Adam, we got it. It's good. And part of our goal here is to make tracks so that we end up at a depth of
1400.
1,400. So we've got a good 300 meters to go. No zoanthids on that. That was interesting. Right?
Yeah.
But I'm just warning you that and the sooner we pull out, stop video. I'm not gonna video the way up because it's gonna take a little bit of time to transfer. But that's for me.
Has Adam introduced himself?
I don't
Chris.
know.
Chris, I got you. Introduce yourself.
My name's Chris. ROV pilot.
Where are you from, Chris?
I am from Medford, East Florida.
Awesome. I feel like a radio DJ. What do you do for a living?
You see it?
how'd you end up here doing this work?
This particular job or this?
That's what. Either way, just what's your pathway? Where you
I
been?
was an electronic technician in the United States Navy, so that gave me a background in electrical clearing application, which
Well,
made it made me capable of what are we doing? Oh, we are? Okay, right here?
Hey, John Ruby. You got
whatever it is, 100.
whatever you need. Okay, all right. I'll try to stay on it. Yep. Sounds good.
oh well.
That was me.
I just don't see how
Thumb slip happens once in a while.
Thumb slip.
Thumb slip. But it's a different oaf here, right? And there's no zoanthids. It's a little paraguay.
Yeah. It's a good collection. It is. That's a good one.
I think it is.
I'm being serious.
I know. All right, we're gonna push in here.
Oh yeah.
So we actually may be calling the dive here, folks, pretty soon. We're halfway. I think it's pretty small, so
Yes.
that's fine. Yes.
It can go in a quiver.
A quiver is a great place for that. So the paragorgia down on the bottom left.
I know
you see
that little baby one. Oh, it has a baby oaf. You
I
see
know.
that?
That's what I'm gonna. And I want to get a little profile on this.
You wanna-
I'm coming in. Just gonna try to get. Do you have my focus?
See how you have the hours to do that, even if you say pull
Oh,
out
I need to
and
take a
there's
sample.
pull out and go back, like, yes, it's not as long,
me?
and so there's not as much.
There you go.
Like, I guess you can get a check in.
A bit hot. Can we come down a little bit on the?
If they've got enough power to get front of the vehicle on.
That's
There you
a
go.
Russ question.
Yeah.
That's another oaf, and it could be a depth thing with this oaf. Yes, it's the same oaf that's down here.
I mean, it's gonna be a bit.
Brilliant.
You get in where we went.
That's fine. That's in focus. That's in focus.
It's just
You're
a
good.
shallow-
You got it. Hold
Yep,
it.
I got
Ready?
it.
Ready?
Not-
You can go down a little bit and just.
Example, you play, and then if it is good, always hang
At
out.
least
Kill
you
the
can try to
rest.
put yourself back down on that wall that you left yesterday, and then.
That's not the same oaf, right? We've been sampling that oaf.
The goal is to try to do.
I don't know.
One more second here, because I want to really see this. That's a humidity. We've seen that a couple times. Some other more polyps down below here.
That's a corral.
That's just not what, that's the first one.
Or maybe not.
Totally fine. I'm just saying
Yeah.
that's-
All right.
Let's sample it.
Let's go. We're ready. Really?
Yeah, Lisa. People are asking about the ROV and if animals try and eat it. It's a really big. It's like the size of a car, so
Oh.
it's not something that many animals would be able to eat or be interested in, but Lisa brought up a time that a grouper used D2 Okeanos Explorers ROV as a place to hide under cover so they could eat a shark. I was on board for that expedition, and it was honestly a really insane thing to experience. So they definitely don't necessarily prey upon the ROV, but they sometimes interact with it.
Transit, going to transit is not
The whale shark would be our friend.
Oh yeah, Randy asked you what the coolest thing I saw. I forgot what the whale shark.
What's that?
That's probably the coolest.
The whale shark?
Like, see what you transit through a current, transit through the current, and get back to the seafloor.
It was cool.
Ceiling of the current, move up to the shallows.
It wasn't a coral. That's not the coolest thing you've seen.
Would you rather go shallow? In that case, would you rather do the shallows part first and then pull
I'd rather go deep shallow, to be honest.
You know,
You're using grip.
yeah, well, that's what we're talking about. So you basically you do 2 dives, do the shallow one first. You go from the deeper, so whatever,
Yeah, deep shallow,
120 with the shallow, and then go back on the position, ship out for the 400 meters, drop it back.
Anything else you want to cover all this?
Huh?
What's the water temperature? The current water temperature is 2.3 degrees Celsius. And seawater freezes at -4, yeah?
Yeah.
So, it's cold.
It's a little out of focus.
That's the question, is transiting through the midwater easier when you're going
They've got the camera
Oh,
controls
okay.
over there.
There is a recording of that grouper shark video on the Okeanos Explorer YouTube. It's called "Oh My Grouper, Look at That Shark."
Oh my god. Oh, nice.
Great.
Okay. I don't even know what sample number we're on here.
Um,
253?
So 253. Yeah.
400 to 250 or the 150 to 0.
I mean, to be honest, logically, you just go from 450 up,
Yeah,
and you go to the next one.
but it's just
21,
daylight as well. How long
253.
is to provide dives?
I will take the full day and then,
Right.
What time is the daylight?
Which quiver are we going in?
It's gonna be like something
I think it's 15.
bit dark by then. So that's what I'm saying. If you did the shallow bit,
Because that's the one that's off, right?
More time.
Thank you.
You run out the dark for the end of that.
For the deeper one, I'll
Yeah,
share.
that's the only reason.
Just point it out then. Let's do the first shallowest shallow dive, and then we'll do the second shallowest dive.
There's no guarantees that we'll go after sunset. It'll just depend on how close we get in. But we've got the option then to say
At least
we
you
stay
won't
in.
have the same problem of being too close to them.
Nice.
There we go.
I mean, and it'll also tell us straight
Awesome.
away if we can actually do the shallow dive.
Yeah. Quiver 15?
the only, yeah, and so I think that.
Yeah, the RV group built these nice collection boxes.
Adam built those.
Adam built those.
Adam is all.
How was it, Adam? Was it difficult? Was it?
The handles are what made them. We played with different handles.
Yeah.
Sometimes if they have poor handles, they're hard to grab in certain positions, but
Right.
we played with this aircraft cable and kind of found handles that
going to,
self-support themselves upwards.
It's nice.
I'm gonna grab the 8 with the other arm so that I can get that green out of the way so that's a little better.
You're probably better off.
Just push it.
Last time I just pushed it in and it just popped out when I, like if you get it in a little bit and then you can
It is. If that handle were longer, it would be sort of dangly and make it
Yeah.
real less
Well,
easy to do it.
We've worked with quiver containers over the years that
Yeah.
all have different handles and different tops. So it's nice that the team has taken time to really test out different things and find what works well.
Yeah, and I remember when I was a graduate student, I built boxes like these quivers, but they were the black part you see there, the
Yeah.
rubber part. I may have was much more sculpted. And
That's what
bigger
we
for me.
still use.
Yeah, and no matter how the pilot puts it in place, it always sort of sits.
Yeah, it like
And then just have to push, kind of, sort of like that, right?
I like these metal tops though. I think they work really well.
Yeah. Yeah, I think PVC is what we use in other. Anyway, it's a great job, Adam. Okay, so
What do we? I guess we're just gonna keep going until we hear.
we're gonna keep going until we hear otherwise.
Okay.
Yeah. Glad that was solved. Oh, dear. So I think we've got maybe 5 or so more minutes left in this dive. We decided to pull it for going for a shallower dive here on Baker. It may be a continuation of yesterday's dive, where we ended a dive and seeing a really massive, unbelievably beautiful wall of highly diverse corals. And then we came over the hill at the top of that and saw a field of more corals loaded with little stars. Weren't they? Those little white ones that
They're really small, yeah. They're really
yeah,
small.
look like stars in the sky, right on the coral. I mean, they do. I mean, I'm just saying. Yeah, that's how they got their name. Little stars.
Brittle stars.
Yeah.
I think it's the 5 arms.
Another crinoid that we saw way deep at the beginning of the dive. Never seeing that.
So we turn up, your DCB's right, you can see, you can obviously say you go, oh, we're feeling it, we can come up, and then you can just go in straight in
So,
the deck.
Yeah.
We're just staying on
Plans are-
bottom
yeah.
to make sure this is dive is conclusive.
Plans are currently being developed for what's going to happen next. This kind of happens on the fly when we
The nice thing about doing this first is,
are working with different currents and different locations.
no distance.
So we want to make sure that we get in the water in the shallow areas while the currents are laying low and the winds are low, so that we're not in jeopardy of being in too shallow water for the ship to navigate.
When we dive so close to the island of Baker here on the equator, we have to really want to dive during the day. So because we're so close, if there's things shift, there's actually several shipwrecks around Baker that were caused because of weather popped up quickly and drove the ships inshore. Put them on the beach. So here we go.
Okay. So, this has never a dull moment around here. If you are watching, we are going to surprise you because we have a limited amount of daylight and we're going to try to do something kind of amazing. So, what we're going to do is end this dive right now. We're going to pull up and then we're going to move back in on Baker, but at a very different depth range. So, in about 2 hours, 2.5 hours, we'll be back on the seafloor. First, we're going to start at around 150 meters and work up to as shallow as we can, and then at the same exact spot, without bringing Sebastian back on deck, we're going to in water fly down and start at around 450, exactly where we were last night, and try to see our PRIMNM
Oh,
reports.
nice to record
I mean,
with.
that's not quite what you said.
Okay.
Yes.
Can we get the currents real fast, please?
Yeah, I think that's our plan. So,
350, it starts to build.
And just change your center, I'll switch it back.
you guys get a 2.5 hour break while we.
Well, we have to process the samples that come
Not
up.
you.
Good?
The people.
Oh, the
online.
people on shore.
We don't get it.
Considered off.
Never a dull moment around here.
No, not yet.
I'm not saying you guys have dull moments. I came out wrong. I'm still tired.
There's a lot of people in this room right now.
So, all right. I think we're going to
No
Should we
associates.
hit the,
head out.
Yeah.
Okay.
and this.
So,
So these guys know? Adam knows. Do you want
Does
to
somebody have the,
start thinking about setting up
you
for
have the lat long of
recovery?
left yesterday?
yes.
As we go? Okay.
Steve has it.
Has what?
Has the lat long of the forest. He wrote it
That's
down.
nice.
Oh. Did I write it down in the notebook?
And
I have it on
There's
my
also
phone.
a-
J-Rod
iPad.
has it.
I have it right
and
here, too.
Someone else has it.
Wait, hold on. Give me a minute.
Enjoy the view of this Davidic gorge here.
Here it is.
Coral.
Lat long. Here you go, Debs.
It's pretty thick central axis, isn't
Yeah,
it?
it's nice.
Right? It's not.
Yeah.
Okay. All right. Anything else you want to?
No, I think we're good.
It's just gonna do that.
Make it happen. This is a great plan.
Okay. All right. We're
Seems like we.
The fans approve.
We're very glad that you approve. In the.
the giant feather underwater.
So it was this edge of this wall.
Yes.
Tim is not from Texas. Tim is from North Carolina.
Yeehaw! Go Tar Heels. I'm all the way. I'm all the way out. Look at that.
Yeah.
It's a monster. We'll just have you look at that.
Yeah. As we
Okay.
fly away.
Oh, absolutely, I'll put you down.
Okay, we only have, so, let's see.
Yeah, what do we have for samples today?
These are rocks. These are, right now?
Yeah.
See how that's done?
Yes.
Okay. So we have 2 corals to process. What's over here?
Slack everyone and tell our
That's
team.
your oaf that was on a rock.
Oh, it's a suction sample.
Yeah.
Roger that. Okay. Surgical.
What?
Yeah.
Yeah,
Yeah,
large.
that's fine. Yeah.
It's gonna be what
So,
number, Tim?
Someone took the notebook. You got it right. You got it right. I should log it.
Or
Yeah.
I just need to know what the, so 254.
Oh, here, you can have this back.
Okay.
Thank you. So, what is this about? What? Okay, we are taking a water sample. What are you doing?
That's my cup in the little chamber.
Stay on
We can do
sea
better
log,
than
don't
that.
worry
We'll-
about the.
we'll
We will, we
Don't
will.
worry about YouTube, I'll take care
Okay.
of that.
Yeah, but I was as soon as they went off bottom, I was gonna do the log off bottom and then turn off DVROs and start that going.
Yeah.
Time is 1918. Depth is 1850 meters. And taking.
Did we fire number?
Number 1. Same with the Yes.
We have title tells here. So
Oh,
number
yeah,
1 just
okay.
fired.
Just the one?
Just the one's fine.
And that is FK 21-254.
Correct.
Did you want to sample this or anything?
No.
No, we're good. Yep. Bring it home.
Bring it home.
All right. So we wanted to image your cup, right? Did you?
Yeah,
it
but
was
we'll let
image-
them get up.
Right. Roger that. All right, everybody. We're off the bottom.
We'll see you.
Off the
We'll
bottom.
see you
Time
sometime
is
soon.
1919.
2 hours.
We'll-
2
we'll
hours
be-
to 3 hours
we'll be
from
right-
now.
So
We'll be
just leave DVR.
back in the water.
Logging,
So what's that saying?
leave DVR logging.
Yeah, so all DVR.
Hey, thanks everybody for watching. Come back in a couple hours. I'll make a YouTube, Facebook. It will go back live. So enjoy your break, and we'll see you shortly.